Okay, so welcome everyone to the 53rd lecture of Dr. Hydri Step 1. Before we begin, can we make sure that our voices are clear? Can you guys hear my voice? Okay, very good. Can everyone hear my voice loud and clear? Okay, very good. So first and foremost, hope you guys are doing well. Did you guys get a chance to solve some UWorld questions from biochemistry like how we asked you to, especially from genetics? Okay. Before we jump into today's lecture, I would like to do a quick revision and recapitulation of previous topics, that is from yesterday, before we begin. So is everyone ready to do a little bit of the revision? Yes? Okay. Do you guys get any chances to read the genetics once again? Although it's not needed because we spent a lot of time studying the genetics portion that we studied yesterday. But did you get some time to practice the questions from genetics or anything? Okay. So let's see how much we can, how much we remember from yesterday's topic, and then we would move forward. Okay. First question: DNA. The nucleosome they consist of 3 to 4 types of histones. What are those histones? The nucleosomes contains 3 to 4 types of histones. What are those histones? Nope. H1 is a linker DNA. H234, very good. H2A, 2B, H3, H4, and H1 is the linker histone. Okay, next one. Next one is which ion is responsible for giving DNA the negative charge? Okay. Okay, which amino acids are responsible for giving histones a positive charge? Okay. Uh, okay, next one. Next one is um, the DNA condensation and histone synthesis occurs in which phase of the cell cycle? S phase. Okay. Which organelle has their own circular DNA? Okay, very good. Okay. Which type of chromatin is transcriptionally active and which type is transcriptionally inactive? Okay. Euchromatins are active, heterochromatins are inactive. What would happen if you methylate DNA? Will it be active or inactive? Inactive. Where would you methylate the DNA? Well, what is the location of DNA methylation? Gene promoters or CPG islands? Okay. 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 Next one is histone acetylation. If you acetylate histone, would it increase the transcription or decrease the transcription? Okay. Active. Very good. Next one. Next one is next one is which type of bonds are the strongest bonds? AT bonds or CG bonds? CG bonds. Okay, good. Okay. Which amino acids are required in de novo purine synthesis? Okay. Okay, very good. Okay. Next one is we will okay. Hope you guys can see my screen over here. Let's talk a little bit about the de novo purine and pyrimidine synthesis over here in a pathway. Let's see how much we remember. Okay, first of all, we have ribose 5 phosphate, will form. Okay. Okay. Which what stands for B? What stands for B? B is PRPP synthetase. Okay. What stands for A? Very good. What stands for C? Okay. D and E. Okay, very good. Then we have glutamine plus carbon dioxide. Right? And then again, okay. Mm-hmm. Okay. A and B. What are A and B? A is carbonyl phosphate, B is aspartate. Okay, what is C? Orotic acid. In the presence of an enzyme. Does anyone remember the name of the enzyme? Although it's not there in the table. What's the name of the enzyme? No, before that, the enzyme required for, very good, dihydroorotate dehydrogenase. That's the enzyme for orotic acid. Okay, very good. Okay, then D and E. UMP and UDP. Okay. Carbonyl phosphate synthesis 2. Where does it come from? Mitochondria or the cytosol? Okay. Where do we find carbonyl phosphate synthesis 1? Okay, very good. Next one. Then we have the purines and the purine salvage pathway, right? We have the purine salvage pathway. The purine salvage pathway, we have the deficiency of two enzymes. So what are those two enzymes? If they get deficient, then the problems can arise. First of all, it's hypoxanthine guanyl phosphoribosyltransferase transferase. Another one is adenosine deaminase. Okay. What's the name of the syndrome where HGPRT is absent? Okay, good. Next one. What is the name of the syndrome where adenosine deaminase is absent? Okay, very good. Okay, can you guys tell me the clinical features of Lesch-Nyhan syndrome? Can you guys tell me the, okay, good. First of all, we have aggression, self-mutilation, then orange diaper color, intellectual disability, gout. Okay, very good. What are the treatments for Lesch-Nyhan syndrome? Macrocytosis, very good. Dystonia, okay, good. Allopurinol and febuxostat. Okay. And what is the name of the What would happen if we have adenosine deaminase deficiency? The main problem of SCID. What is the main issue with SCID? Good. Lymphocytotoxicity. Okay. Okay, good. If you have a single nucleotide mutation at the third position of a codon, will it change the entire amino acid? Yes or no? Why? Because it's the wobble position. Okay, good. Next one. Next one is there are so can you guys tell me the organelle whose genetic code has been the same throughout and the entire throughout the entire evolution? Mitochondria. Very good. Okay. Okay. Which enzyme has 3' to 5' exonuclease and 5' to 3' synthetic activity? Okay. Which enzyme is responsible for the degradation of RNA primers? For degradation of RNA primers and replacing it with DNA. Okay. Which enzyme is responsible for joining the Okazaki fragments? Okay. Which enzyme is responsible for unwinding of the DNA helix? Okay. Which enzyme is responsible to remove the supercoils of the DNA helix? Okay. Which enzyme is responsible for adding primer to initiate the replication? Primase. Very good. Okay. And what are the functions of the single-stranded binding proteins? Very good. Okay, you guys are doing exceptionally well in genetics. Okay. Have you guys studied genetics before, or was this the first time? This is the first time in school. Okay. Okay, so this is exceptional how you guys can answer everything from the things that we have discussed yesterday. Are you guys looking at the book and then providing the answers? Okay, good. Okay, so thank you so much for retaining the informations which we have provided for you. And hopefully the lectures are helping you guys out to remember most of the high yield things. Was it helpful, the lecture on genetics? Okay. Okay. Do we have any new students over here with us today? Do we have any new students who are here with us today? Do we have any new students here with us today who has joined the lectures and this is their first this is their first time doing the lectures with us? Okay, I guess not. Okay. Okay, so if you are a new student, basically our lectures are a 4 to 5 hour lecture. The first half an hour or to 15 minutes to half an hour is done doing the revision and recapitulation of the previous topics, which we are done. Now we are going to begin the second part of the lecture, that is reading the first aid step by step, text by text, trying to understand, master, and trying to retain the high yield information so that we can do good in the UWorld question banks. Third part is we discuss the UWorld and emboss notes. And the fourth part is we do UWorld or Kaplan question banks. That's that. Fifth one is if anyone has a question, they can ask me questions regarding the lecture, transactions, or any other process. That's that. Okay. So without further ado, we're done with the first segment. That is revision and recapitulation of yesterday's topic. Now let's begin with the second part. Is everyone ready with their first aid? We will begin with the text. Okay, we will begin with page number 39 from 1st/8/2020. Okay, where we will be talking about mutations. We will be talking about mutations. Okay, so let's begin. What we are going to do is we're going to read, and then we will use the blank page to make sure that we have understood everything. Okay, that's that. Before we jump into mutations, first of all, we have to understand the structure over here. Okay, we have to understand the structure of the amino acid. If we have to understand the structure of the amino acid, let's for 1 second, let's just go to our blank page and see what we're dealing with over here. First of all, an amino acid, they are multiple codons, right? So we have an amino acid over here. Okay, we have an amino acid, right? Okay, so let's say that this is the 5' end, this is the 3' end of the amino acid, and then over here, every codon will code for one specific amino acid. Okay, for example, if we have one codon over here, then one codon over here, one codon over here, one codon, and so far and so forth. Okay, over here, let's say that the codons are G, A, G. Then we have G, A, A. Then let's say we have U, G, G, and then A, G, A. So on and so forth. Okay, so that's exactly what it is. So basically, the fact that G, A, G, these codons, that is guanine, adenine, and guanine, these three codons are coded together. When they are read in one codon, this part of the DNA will code for one specific amino acid. Let's say, for example, GAG is very well known and very famous for glutamate. Okay, at the same way, you remember yesterday we talked about AUG. AUG is universally known for methionine. That is also an amino acid. Okay, that is also an amino acid. And then we also have our UAA, right? These are known for the stop codon. So basically, if you had a directory book, if you had a book which would have told you all the different types of codons which codes for the amino acid, then you can easily read which codon codes for which amino acid. But there are some things over here. That is, we know that every position of the nucleotides, right, they are important. Basically, what happens is, if you knock out one nucleotide, okay, right now, let's say you knock out one nucleotide for some reason, maybe let's say due to some mutation, this nucleotide is knocked out, meaning that this nucleotide is absent. Okay, this nucleotide is absent. So what happens is you start reading the whole thing like this. You start reading the whole thing like this. G, A, G, because this G comes to fill up this place, and then AAU, so AAU, and then since AAU is now another codon, then GGA becomes another new codon, GGA. So what happens is, do you guys realize that before you could have had one type of amino acid, but right now, since one codon is knocked away, the entire sequence has changed. Right now, instead of forming glutamate, this can form exactly something else. This can form GAG. For example, GAG is still, for example, let's say GAG is still glutamate, but then again, you have AAU. AAU will code for an entirely different amino acid. AAU will code for an entirely different amino acid. So the fact that this has happened, this is known as mutation. But the thing is, usually, if you knock out the third codon, this position is the wobble position. As we have discussed before, this position is the wobble position. So what happens is, even if this position gets knocked out, there is a very low chance that the DNA will express any other amino acid uh, except the one it was supposed to express. So usually it's not a problem, but the main problem arises when the mutation occurs in this position or in this position. Okay, so that's that. So that is basically what we have to understand from over here. Okay. And so right now we can jump into the lecture. First of all, mutations in DNA, the severity of the damage, meaning that the severe mutation, the extremely severe mutation is frame shift mutation. After that, you have nonsense mutation. Then you have missense, and then the least severe or the least harmful is silent mutation. It's silent mutation. Next one. Next one is if you have a mutation where the mutation occurs from one purine to another purine, this is known as transition. Transition. If you have the mutation from one purine to another pyrimidine, this is known as transversion. And we know the purines are pure as gold. So A and G. And cut the pi. So we have CUT as pyrimidines and A and G as purines, right? So if it's between purine to purine, it's transition. If it's from purine to pyrimidines, it's called transversion, okay? This is not very high yield. You do not really have to remember this as much. That's that. Next one. Next one is the mutations. Right now, we will talk about the mutations exactly like the one we talked about over here, single nucleotide substitutions. Single nucleotide substitutions. First of all, we have three types of single nucleotide substitution mutation. Out of them, we have silent mutation, missense mutation, and then we have nonsense mutation. Silent mutation. Let's talk about the silent mutation first. Okay. So once again, this is the 5 prime, this is the 3 prime over here, and then we have the amino acids. Okay. We have the amino acids. Silent mutation. Silent mutation especially means that if there is a mutation, the nucleotide substitution for the code will still code for the same amino acid. It will still code for the same amino acid even after the mutation has taken place. So if I tell you that the silent mutations, they will still code for the same amino acid, is there a possibility that the mutation is occurring at the third position or at the wobble position? Yes or no? Yes. So silent mutation is basically the mutation that is occurring in these positions over here, the wobble positions. The third positions or the wobble positions. Okay, so even after the mutation occurs in these ones, the amino acids will still code for the same thing. That's that. There will be no shifting. Okay, next one. Next one is missense mutation. Missense mutation is basically the nucleotide will substitute, the nucleotide substitution will result in the changed amino acid. So what would happen is in missense mutation, for example, there will be a nucleotide substitution. Substitution meaning that there will not be knockout of the mutation. The nucleotide will be substituted. For example, over here we have GAG, right? What if this G was substituted for C, right? Now we have the new type of mutation that is GAC instead of GAG. Before it was coding for glutamate, now it will code for entirely another amino acid. For example, GAC is known to code aspartate. GAC is known to code aspartate. So over here right now, it was supposed to code for glutamine. Right now for this substitution, okay, missense is substitution. Silent does not matter. Silent, it does not matter. Okay, missense is substitution. Substitution. Okay, there is no knockout of genetic code. It's just one nucleotide is substituted for another nucleotide. So right now it will code for another thing. Next one is nonsense mutation. And nonsense mutation, what happens is we have the nonsense mutations which are responsible for causing an early stop to the DNA synthesis or to the amino acid synthesis. So what happens is we have the stop codons, right? Stop codons are U are away, U go away, and you are gone. If any one of these codons appear by mutations over here, then what happens is, for example, this is UAA. This should not have been here. Okay, this should not have been here, because if the stop codon was already here, then the DNA would have stopped. So, for example, let's keep this something like UCC or like or whatever. Okay, but what happens is, let's say that for some reason there is a nucleotide substitution. Once again, if there's a nucleotide substitution for which, for example, this is UAC. Okay, UAC is still another codon which is coding for another amino acid. For example, let's say that this C right now is substituted for G. Then what would happen is the amino acid synthesis would keep on occurring in this way, and then they would come across this stop codon, and then the amino acid synthesis would stop right over here. So this amino acid will not form. So what this will result is, this will result in an entirely different protein. So nonsense mutations are, nonsense is early appearance of stop codon. Early appearance of stop codon. That's that. Okay. Now let's read the text. Let's see what the text says. So we have silent mutations, which is basically the nucleotide substitution codes for the same amino acid. The base is usually changed at the third position or of the codon, or the tRNA wobble. Basically, the questions will usually come from this side. The answers are the names, okay? They will ask you whether you have the knowledge to differentiate silent mutation from missense mutation and from nonsense mutation, okay? So if you have to test yourself, cover this part with your hand, read out this part of the text, and see whether you can remember that this mutation, the name of this one is silent mutations. Okay, that's the best way you can test yourself. That's that. Next one is missense mutation. Missense mutation, the nucleotide will be substituted. Nucleotide substitution will result in, okay, the nucleotide substitution will result in changed amino acid. The entire amino acid will be changed for the substitution. If the amino acid, if the new codon which has been substituted, if it has the same properties as the previous codon, then the new amino acid will have the same similar chemical structure. Or, for example, in case of sickle cell disease, we have the substitution of glutamine with valine, of glutamic acid with valine. That's what results in sickle cell disease. If glutamic acid with valine, if that did not happen, then the RBCs would not have had those sickling features. They would be normal RBCs. Since there is a missense mutation, then that's the reason why we have sickle cell disease. So next time, if you will receive a question, for example, sickle cell disease, you might receive a question, sickle cell disease, what type of mutation occurs in sickle cell disease? And your answer should be missense mutation. Missense mutation. This is a high yield question. You will receive this. Okay, the answer is missense mutation. Please write this down over here so that next time when you go through first aid, you can easily review this. Next one is nonsense mutation. Nonsense mutation is basically stop the nonsense. Stop the nonsense would make you think that there's an early onset of amino acid synthesis, which stops because stop codon appears out of nowhere. That is UGA, UAA, and UAG. Okay, that's that. So these are the three types of mutations which we have talked about, which are the single nucleotide substitutions over here. Mutations are extremely important. Okay. Next one. Next one are other types of mutations. Okay. So there are some other types of mutations. Okay. First of all, we have frame shift mutation, which is the worst kind, and then we have splice site mutation. Frame shift mutation is basically if you delete or insert a number of nucleotides which are not divisible by 3, this will result in misreading of all nucleotide downstream. The proteins may be shorter or longer, and its function may be disrupted or altered. So what happens is when we have additions of nucleotides, right? For example, Okay, for example, what happens is, let me break this down for you. Okay, so for example, over here in a normal DNA structure, what we have is we have pairs of threes, right? For example, we have 1, 2, 3. So the first three are, for example, this is G, A, G. So this is 1, 2, 3. Then we have 4, 5, 6, right? At the fourth position, fifth position, sixth positions, and then we have A, G, U, or and so far and so forth. This keeps on going on. 7, 8, 9, 10, 11, 12. Okay. So so in a normal amino acid without, I mean, in a normal protein, all these amino acids, they are arranged in a structure like this. So what happens over here is that if you try to divide the amount of nucleotides, for example, over here you have 12 nucleotides, right? If you try to divide this number by 3, okay, so 3 times 2, 3 times 4 is 12. So the number comes to an equal 4. So, this protein has the nucleotides coding for the amino acid, which can be easily divided by the number 3, and it comes to a solid number. So there is absolutely no mutation going on over here. What if there is a mutation that you have the 5 prime, 3 prime, right? You have first 3, second 3, third 3, and then over here you only have 2. So what happens is you have GAA, UAA, my apologies, UGG, then you have ACG, and then you have AG, and then the last one is not there. So what happens is you have 3, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. You have 11 nucleotides. So this number over here, 11, if you try to divide it by 3, this does not come to a solid number. It's a decimal number. So what happens is this results in misreading of all the nucleotides. This results in misreading of all the nucleotides. So what happens is over here, if there's any mutation which results in either a deletion of a nucleotide, or example, instead of this, for example, over here, let's say you had 10, 11, 12, okay? And then you had insertion of another nucleotide over here. So instead of having 3 codons, now they have 4 codons. So now the number is 13, right? So you have 13 divided by 3, and then the number is still not a solid number. So whenever you get the nucleotides which cannot be divided by 3, you can conclude that this patient or this subject has a frame shift mutation where the entire frame is shifted. So what happens is over here right now, let's say that right now, let's say that when you have this codon with, if it's deleted, right? If this is gone away, so the entire frame of the codon will be shifted. So right now the readings of the amino acid will change, because what would happen is, for example, if this one goes away, this one will come, and then this one will form another amino acid, and then so far C, G, and A will come, and they will form another amino acid. So there will be misreading of all the nucleotides. This is known as frame shift mutation. The easiest way you will receive questions in frame shift mutations is that they will provide you with numbers. Whenever they provide you with numbers of nucleotides, for example, they tell you that they have a mutations over there, and then over there they have 52 nucleotides. With the calculator, try to see if 52 is easily divided by 3. If 52 is divided by 3 and the number comes to a solid number, then there's no frame shift mutation. If 52 is divided by, if it's not divided by 3, then high possibility there's a frame shift mutation. Are we clear about this? Yes or no? Easy to understand. Frame shift mutation. Question. What is your question, please? Okay, can you show what you drew about? I thought adenine, etc. Are bases. They are not nucleotides. We're talking about nucleotides, adenosine, guanine, right? That's the one. Okay. Are we clear? Okay, nucleotide bases. Okay. Are we clear about this, Dr. Karbasi? Adenine, guanine, cytosine, and thymine. Okay, we're talking about the nucleotides. We're talking about the nucleotides. The nucleotides are basically the nucleotides. They're basically your bases. Everything is a base. To be honest, it's a base with either deoxyribose sugar or it's a base with phosphodiesterase bond. So if it's a base with phosphodiesterase bond, it's a nucleotide. If it's just a sugar plus the base, then it's a nucleoside. So if you say adenosine, you're talking about nucleoside. If you're talking about adenine and guanine, you're talking about nucleotides. Are we clear? So you think so you said that I thought adenine, etc. are bases. They are bases. They are nucleotide bases. Okay. Are we clear about this? They're nucleotide bases. Okay. Next one. Next question was from Dr. Mohammed. Can you show what you drew about missense mutations? Missense mutation is basically this one over here. Missense mutation is what happens is you have a substitution of one nucleotide with another substance and with another nucleotide, and then the entire amino acid is read in a completely different way. That is missense mutation. That is missense mutation. Okay. Next one. Next one is can frame shift mutation caused by deletion of a third amino acid, as you showed? Frame shift can occur because of a deletion or an insertion, either one. What would happen is if the codons are not divisible by 3, that's how you can identify a frame shift mutation. And then the entire genetic code is read in a completely different way. That's that. Any other question? Any other question? What is a frame mutation? We just talked about this. We just talked about the frame shift mutation. What happens is you either have a deletion or you have an insertion of a genetic code that results in the misreading of all the nucleotides downstream. So what happens is if the genetic codes are not read or they are not divisible by the number 3, then you can identify that this codon is a frame shift mutated codon. Okay, and this would result in a different amino acid. That is a frame shift mutation. Okay. Why are you reading 5 to 3? That the DNA is read from 3 prime to 5 prime. That's not what we are focused on, the reading of the DNA. Do you understand? We're not trying to read the DNA. We're trying to show you that when you have 5 prime and 3 prime, and then you have the codons over here, that any type of mutations can change the entire codon. That's that. It does not matter if I'm reading it from 5 prime or 3 prime. That's not what we're talking about. We are talking about the mutations of the nucleotides. Okay. Okay, next question. I think you misunderstand. If my third codon position got deleted, will it be silent or frame shift? If your third codon position got deleted, and if it's at the wobble position, then it's a silent mutation. Okay, if it's at the wobble position, it's at the silent mutation. But what happens is that in frame shift mutation, let's say that over here, as you can see, that in frame shift mutation, if you have an insertion in between the wobble position, in between the position, for example, this is the wobble position. If you have an insertion in between this and this, then you have a new type of amino acid. Or if you have a deletion, right? For example, if at the wobble position, this gets deleted, then what would happen is usually this should be a silent mutation. But if this gets deleted and this gets shifted over here, right, then you have an entirely new codon for coding for an entirely new amino acid. So that's how you have the frame shift mutation. Okay, so it's very hard to say whether the deletion at the third position can cause frame shift or not. But most of the time, if you have a deletion which is a single mutation and there is no shifting of codon, what happens is over here, let's say you have a knockout of this and there is no shifting, that this G is not shifted over here. So this is just a silent mutation. If you have a shifting, then it's a frame shift mutation. Try to focus on the word shift. Shift means after the mutation, there's shifting of the codon. So that's that. Okay. If there is shifting, then it's a frame shift mutation. If there's no shifting and it's just one knockout, then it will not be a problem. Are we clear about this? Can missense mutation also occur in third position? No, missense cannot occur in third position. In third position, if missense mutation occurs, if there's a substitution at third position, it's called silent mutation. Okay. Okay, once again, we're not reading the DNA from 5' to 3'. Okay, we're just talking about the mutations. That's all, nothing else. Okay, because your example wasn't third. For missense mutation, my example wasn't third. Or for silent mutation, my example wasn't third. Let's see over here. For missense mutation, my example, where is my example? For missense mutation, my example was in the second one. Okay. Are we clear?
Okay, because you changed GAG to GAC, and then you said that's, yeah, that one, yeah, and you said that was
GAG to GAG to GAC. If that was the one, no, then missense mutation will always occur at the second one. It will never occur.
Okay. Thank you.
Okay, you're welcome. Is every third position called wobble or just some of them? Every third position is more or less called the wobble position because it's the silent position where if there is a mutation, then this will code for the same amino acid. Okay. Any other question? Anyone? Okay, good. Thank you so much for asking your question because you have to get rid of all the confusions. Okay, that's that. Okay, very good. Then the last one. So we are done with most of the mutations. Then the last one is, before we move on to the last one, you have two very high yield diseases, which we have already talked about in New World Notes: Duchenne muscular dystrophy and Tay-Sachs disease. Do you guys remember we talked about the fact that how DMD and Tay-Sachs, there are frame shift mutations? Yes or no? Frame shift mutations. Yes, we have already mentioned them. So you have that written over here in the first day too, right? DMD and Tay-Sachs. So please write it down over here. DMD. There are a lot of other diseases with the mutations, but since it's mentioned in the first aid, high possibility you will get questions from over here that they might directly ask you what type of mutation is Duchenne muscular dystrophy. So DMD is frame shift. Tay-Sachs is also a frame shift mutation. Okay. Okay. Next one is splice site mutation. Splice site. Before we talk about splice site mutation, are we well aware of the fact that an mRNA is formed from the protein, from the DNA, and that codes for the translation process of the protein synthesis? Yes or no? Okay. So basically, do we understand that when an mRNA is formed, it contains introns and exons? Do you guys know introns and exons? Introns and exons. Okay. Which one are important and which ones are important and which ones are not important? Are introns important or exons important? Introns and exons. Basically, introns and exons are segments of the DNA. I mean, they are segments of the mRNA. If what happens is, for example, that you have introns and exons, right? If the unimportant segment. Okay, for example, if you have an intron and an exon, the introns are the parts of the mRNA which does not code for any protein. The introns are the parts of the mRNA which does not code for any protein. So what happens is we have to get rid of those introns when the mRNA codes for the translation process of the protein synthesis. So what happens is the first mRNA that is formed, that is called pre-mRNA. Pre-mRNA. After you remove the unimportant parts of the pre-mRNA, you have the mature mRNA. I mean, you have the mature mRNA, which consists of only the exons, and the introns are removed. But for some reason, let's say that we have a mutation, and for that mutations, that intron removal has not occurred properly, right? So you still have the introns in place. You still have the introns in place. What this will do is, if there is retention of the introns in mRNA, the protein structure will be impaired, or they will have an altered function, meaning that the exons, they could have produced a protein with an entirely different function. But right now you have retention of introns in the mRNA, and now they have proteins with impaired function. So basically, this is very easy to understand. Basically, if anyone asks you, what is a splice site mutation? Splice site mutation is basically mutation in the process of splicing, where the introns are not removed properly, and you have retention of the introns, and those retentions of the introns resulted in an entirely different protein with an altered function. What are the examples? The examples are some causes of cancers, dementia, epilepsy, and beta thalassemia. Beta thalassemia is a splice site mutation, Gaucher's disease, and then you have Marfan syndrome. So over here, we'll write down over here, beta thalassemia is a splice site mutation. Gaucher's disease is a splice site mutation. Marfan syndrome is a splice site mutation. Okay, just for the fact that first aid has mentioned these diseases in their examples, there's a higher chance and higher possibility that you will receive questions directly. They will ask you that whether you remember the fact that these diseases are associated with these mutations. What USMLE does at times is they try to see whether you have actually read the first aid properly or not. So that's why they ask you direct questions like this. So that's that. So have you guys understood the mutations? Yes or no? Yes. Okay. Can I give you guys 2 minutes to read this, to read the names of the disease and the mutation which causes them? Okay, because this is actually pretty high yield, especially for AMBOSS, and also to some degree into for UWorld and NBME, is that they can directly ask you what type of mutations are these ones. Okay, I'll just give you 2 minutes. In those 2 minutes, please read this out. I will test you whether you have learned it or not. Okay, so 2 minutes on the clock. It's 9:52. I'll ask you at 9:54. Okay, is anyone done? If you're done, please let me know. Okay. Sickle cell disease. What type of mutation is sickle cell disease? What type of mutation is sickle cell disease? What type of mutation is Duchenne muscular dystrophy and Tay-Sachs disease? Okay. What mutation is occurring in beta thalassemia? Okay, you have a type of disease in where the macrophage, they resemble tissue paper like crumbling appearance. What is the name of that disease? Along with that, the patients, the patients, they also have bone pain, extreme bone pain. Extreme bone pain, macrophages, they appear as tissue paper like crumbling appearance. What type of mutations are we talking about in this disease? Okay. You have a disease where the patients have a higher tendency of having mitral valve prolapse. Okay. It's a connective tissue disorder. What type of disease am I talking about? Especially for mutations we have discussed. Very good. Marfan syndrome. Okay. So it's a splice site mutation. Okay. Did you guys realize that I tried to make the questions a bit tougher, but I still couldn't get through to you? Okay. Still hard to stop you guys though. Okay. What is the name of that disease where over there you have cherry red spot in the macula? Okay.
Tay-Sachs.
Tay-Sachs. Okay. What other disease has a cherry red spot in the macula? Cherry red spot in the macula. Any other disease? Any other disease?
Gaucher crab.
Niemann-Pick disease. Niemann-Pick disease. Okay. Very good. What's the name of the disease where you have the presence of globoid cells? Globoid cells. Nope. It's not Fabry's disease. Have you guys heard of Krabs disease? Krabs disease? Okay. What's the name of the disease where you have, okay, you know what, we're not going to do this anymore. We're not going to go to lysosomal storage diseases. Okay. We're going to move forward. Okay. Are we done with the mutation so that we can move forward? Yes or no? Okay. All right. Let's move forward to the next page over here. Lac operon, very, very high yield for Euro. Lac operon, it seems, how difficult is lac operon? How difficult and complicated is lac operon? Lac operon. Do you guys know what lac operon is? Hard, very hard. It's absolutely not hard. It is the most easiest thing you'll ever study. It is extremely easy. Okay. The problem is first aid does not know how to express the fact that it's easy, because you have to understand that first aid is a guidebook. Okay, it's not a real textbook. Lac operon, what this is, basically what happens is the main confusion happens because in one page we're talking about humans, and then in another page we're talking about bacteria. Okay, that's where the main confusion is. This lac operon is present in E. coli. What happens is that this is a classic example. Lac operon is an example of a genetic response to an environmental change. So lac operon is an example where your genes or the genes of the bacteria, they change according to the change in their environment. Especially if we talk about this lac operon, for example, in an E. coli, the main substrate is glucose for their metabolism. So when glucose is not there, and lactose is there. So when glucose is not there, basically when glucose is there, the lac operon, it functions properly, right? So what happens is when glucose is there, when glucose is not there and the lactose is there, okay, when glucose is not there and the lactose is there, the lac operon will switch to lactose metabolism, meaning that the E. coli, they like to have low glucose, and they like to have high lactose. Okay, that is the main thing over here. So when you have low glucose and high lactose, that is when the E. coli can express their genetic processes, and their the proper metabolism can occur. Lac operon is basically the metabolism that is the genetic process that is required for formation of the amino acids that is required for the breakdown of the lactose. Okay, that is required for the breakdown of the lactose. So basically, these genetic codes, they will code for the enzymes that is required to break down the lactose. But this only has to happen when glucose is not there and lactose is there. Usually, when glucose is there, lac operon is not functioning because they do not have to produce the enzymes to break down the lactose. For example, right now, there is high amount of lactose in the E. coli environment. What they will do is they will turn on the lac operon, and this operon can go and activate the enzymes that will result for the breakdown of the that will result in the breakdown of the lactose. That's that. And this is nothing but just an example. This is just an example of one part of the E. coli. That's that. Okay, so let's talk a little bit about this because you get a lot of questions from over here. I still don't know why they try to make a big fuss about lac operons. Okay, but since they do, important for us because we have to understand this. So what happens is over here, okay, lac operon. So we're talking about lac operon of E. coli, okay, not humans, not humans, E. coli, lac operons in E. coli. What happens in a high lactose and low glucose state? First of all, let's draw the genetic sequences. What happens is when you have a sequence like this, the lac operon, they have certain sites. Okay, they have certain sites. So first of all, the first site of the lac operon, okay, the first site of the lac operon, this site is called CAP, and I would like to think of this as the cap or the hat which the genes wear. Okay, next one is promoters. Okay, gene promoters. We talked about the fact how promoters are important because if you stimulate a promoter, then you have the synthesis which will start occurring. So that's that. And you have the promoters. Then you have the operators. Operators meaning that they are regulators. They will regulate. Okay, they are the regulators. Then you have the genetic codes. Okay, then you have the genetic codes. Usually the first 3 or 4 or the first genetic codes are the initiation code, especially, for example, they are basically the augmenters, or they will start the process. So so the first three codes are in E. coli only. It's lac Z, lac Y, and lac A. Lac Z, lac Y, and lac A. And over here you have a protein. Over here you have a protein. Okay, this protein is known as repressor protein. Repressor protein. Repressor proteins, when they get activated, they will repress the genetic synthesis. I mean, the amino acid synthesis from the genetic codes. So if you have to stop the amino acid synthesis from the codes, you have to activate the repressors. Okay, if you want to initiate the amino acid synthesis from the genetic codes, then you have to block the repressors. That's basically that. Okay, now what happens is we have talked about the fact how there are two conditions that are required for the initiation of the lac operon. The first condition that we have talked about is low glucose. If you have low glucose, what happens is, if you have low glucose or less glucose in the environment of E. coli, if there is presence of less glucose in the environment of the E. coli, what they will do is they will activate adenylate cyclase. And we all know what happens when we activate adenylate cyclase is that ATP gets converted to cyclic adenosine monophosphate. CAMP. CAMP, when this gets activated, CAMP, what this does is CAMP will activate the CAP. CAP stands for catabolic activator, catabolite activator protein. So CAMP will go and CAMP will activate the CAP. Okay, CAMP will activate the CAP. If CAMP activates the CAP, what they will do is CAP will activate the promoters. So basically CAP will activate the promoters, and once the promoters get activated, the sequence can start. That is the amino acid sequence can start. So usually what happens is this is usually not working, or you usually do not need to activate the lactose breaking the lactase breakdown and the lactose breakdown environment, enzymes, right? Because usually what happens is when you have a high amount of glucose, you really do not need enzymes which are needed to break down lactose in your system of the E. coli. But when glucose is not there and lactose is there, so when glucose is not there, immediately when the E. coli detects a change in the environment and it starts the production of amino acids, especially the enzymes which are needed to break down the lactose, because they think that when glucose is not there, lactose is coming, so we have to break down the lactose, so they start producing this. So operon is nothing but an example of an environmental change, and this environmental change is occurring in E. coli. Like operons, we have a lot of genetic codes like this in our body or in our system, where whenever we have an environmental change, that thing will either activate or deactivate our genetic code accordingly. So for example, let's say that we have high amount of lactose or something like that, and our body needs to produce a certain amount of enzyme in a high amount, then in a similar process, our body will function in a similar way. Okay, that is exactly what operon is. It's just an example. Okay, so we have talked about the first one, that is if glucose is not there, you have adenylate cyclase, which activates ATP to which converts ATP to CAMP. This activates CAP. CAP activates promoter. Promoter activates LAC-Z, LAC-Y, LAC-A, and then the enzymes are produced. This is one condition. Another condition is, let's say instead of low glucose, right now you directly have high lactose. Okay, you have high lactose. High lactose, what they will do is they will not activate the CAMP. High lactose will deactivate the repressor protein. The repressor protein usually goes and it binds. It usually goes and the repressor protein, it usually goes and it binds over here. It binds like this. Okay, it binds to the operator. So the amino acid synthesis from the genetic code, it usually cannot happen. But when lactose goes, and when there's high amount of lactose, what they do is the high amount of lactose will go over here. Okay, let me show you what happens. Let's say these are the lactose molecules. They will go and they will bind over here. When they bind over here, there's not enough space for the repressor proteins to go and bind to the operator. So this doesn't happen. As a result, once again, the amino acids are synthesized from the genetic code. So this is operon. That's that. So the fact is, you have to remember two things over here. That is, how is operon activated? Operon is activated by low glucose and high lactose. And how does low glucose activate the operon? By CAMP binding to CAP, and high lactose activates the operon by binding to the repressor proteins. That's that. Okay. Did you guys understand this? Did you guys understand the operon? Operon? Yes or no? Function of repressor protein. They will go and bind to the operator and prevent the activation of the genetic code to prevent the formation of the amino acid. Okay. Okay. Let's read the text. Let's see what the text says. We're not done reading the text. We just discussed this. What happens is lack operon is a classic example of a genetic response to an environmental change. Glucose is the preferred substrate in E. coli, but when glucose is not there and lactose is there, the lack operon is activated to switch to lactose metabolism instead of glucose metabolism. This sudden switch of glucose breakdown to lactose breakdown is a classic example of an environmental change. And this keeps on happening in E. coli and a lot of other types of organisms. That's that, even in humans. So for example, you have low glucose. What happens is in low glucose, you have increase in adenylate cyclase activity, which will increase CAMP from ATP. And CAMP from ATP will go and bind to the cap site. And then once the cap site is activated, this will cause amino acid synthesis from the genetic sequences. So increase of transcription. Either there could be low glucose or there could be high lactose. High lactose, well, what they would do is they would go and directly go and bind to the repressor protein. First of all, repressor proteins are the proteins which usually stays bound to the operator region, preventing the amino acids to be formed from the genetic codes. But when lactose is there, it goes and binds to the repressor protein and it inactivates the repressor protein. Accordingly, what happens is you have the proper functioning of the lac operon, and the enzymes are produced from the genetic code. That's that. So high lactose will go and unbind the repressor protein from the operator site. They will remove the repressor protein from the operator site, and this will increase the transcription. Okay. Are we clear about this? Are we clear about this? Okay. Okay, now let's talk about the regulation of lac operon, from where you get the extremely hard questions in UWorld. This is extremely easy, okay? So what are the situations when lac operons will be extremely stimulated or not stimulated or stimulated to a very low amount, okay? First of all, if you have low glucose and high lactose, okay, what would happen to the lac operons? Will they be strongly stimulated or strongly expressed? Yes or no? Yes or no? Okay. So they will be very strongly stimulated. Okay. What would happen if you have high glucose, no lactose? What would happen to the lac operon? Obviously, the exact opposite will happen, will not be expressed. Okay, next one. Next one is if you have low glucose and lactose is not there. Lactose is not there. So what would happen is if lactose is not there at all, there's absolutely no point to form the enzyme that is required to break down the lactose. So even if glucose is low, but lactose is not there, the lac operon has no need to form the enzymes to break down the lactose. As a result, the lac operons will not be stimulated. Okay. And then if you have high glucose and lactose is there, but not in extremely high amount, it's just there in the normal amount, then what would happen is the lac operon will be stimulated, but the lac operon will be stimulated in low amounts. Okay, are we clear about this? Okay, next time you receive questions from lac operons, you will receive them either from the regulations, from the function, or from the identification. They might try to ask you to identify, so that's that. Okay. Is everyone clear about this? Any questions? Why low amount? What? Okay. Okay. Write this down. This is high yield. Okay. This is pretty high yield. Write this down. This is an extra piece of information over here for lac operons. I'm just going to write it over here. You guys can make a note over there. Okay. lac Z, that is this one, when this gets stimulated, they will code for the amino acid or the enzyme beta galactosidase. Okay. Then lac Y, when this gets stimulated, they will code for the enzyme permease. It's not here in the first aid, but it's mentioned in New World. If it codes for, and then once it codes for lac Y, lac A, this will code for thiogalactoside transacetylase. Okay. Lac Z will code for beta galactosidase. Lac Y will code for permease, and lac A will code for thiogalactoside transacetylase. Okay. Are we clear about this? Did you guys write them down? Okay, next enemy. Okay, isn't enough glucose there? It's there in low amount because we need to break down the lactose, not the glucose. That's why it's called the lac operon. So even in the presence of high glucose and normal amount of lactose, very low amount of lac operon will be activated. That's why. Okay. Beta galactosidase. Then lac Y is permease. Then lac A is thiogalactoside transacetylase. Oh, my apologies. Last one, transacetylase. Okay, transacetylase. Can we move on? Okay, this is just for your knowing that these are the enzymes. You might not be tested on this, but if you are, just need you guys to be ready. I do not want you guys to receive new information. So just providing the information you might receive in UWorld by itself. Okay. Okay. Is everyone ready to move on? Is everyone ready to move on? Yes or no? Okay, good, very good. Let's move on. Do you guys need 1 minute to learn the names of the enzymes from the from the genes? Is why it's taking you guys some time to answer? Okay, how about I give you guys 1 minute to learn the names, because I know it's a bit difficult. Lac Z, lac Y, lac A. Lac Z stands for beta galactosidase. Then lac Y stands for permease. Lac A stands for thioglycane transacetylase. Thioglycosidase transacetylase. Okay, I'll give you guys 1 minute. What does lac Z stand for? Lac Z. Lac Z, what does it stand for? Beta galactosidase. Lac Y. Permease. Lac A. Okay. Okay, good. Okay, next one that we want to go and talk about is DNA repair. DNA repair is extremely high yield. Okay, let me tell you how you will receive questions from DNA repair. Okay, first of all, the best way you will receive questions from DNA repair is not from over here. They will tell you that you have a patient with xeroderma pigmentosa. What type of DNA repair is taking place in a patient with DNA with xeroderma pigmentosa? What type of DNA repair is absent? Okay, so for example, that if you have a defect in nucleotide excision repair, this can result in xeroderma pigmentosa. So the answer is nucleotide excision repair is absent or it's not working. So this is the best way you will receive questions. So your questions will come over here from the names of the diseases, and your answers will lie in the names of the repair and whether you have the knowledge to differentiate one type of excision repair from another type of excision repair. So with that being said, let's begin with DNA repair. First of all, you have single-stranded DNA repairs. Then you have double-stranded DNA repairs, especially being as simple as, for example, this is one strand of the DNA. This is another strand of the DNA. If you have repairs in this strand only, then it's single. If it happens at the same time in both strands, it's a double one. Okay. Then the first one is nucleotide excision repair. Nucleotide excision repair. What happens is you have specific endonucleases. Okay, endonucleases. What do we mean by endonucleases? What do we mean by endonucleases? Endonucleases. What does the word nucleases mean? Anyone? I need some participation from you guys, please. If I want to finish this, either you know it or you do not. Yes or no? Okay, good. Cutting. Exactly. Cutting or lysis. That's exactly what I wanted to hear. Basically, meaning that you have specific endonucleases which will release the oligonucleotides, which will contain the damaged bases. For example, if you have, damaged base, right? For example, you have GAA, AGG, or so far and so forth. Over there, you have one nucleotide which has been damaged, especially from, let's say, you have sunlight, right? The sunlight has, this is just an example. For example, sunlight is high in ultraviolet rays. The ultraviolet rays, what if they go and they damage one of the nucleotides that's there in the DNA of your skin, right? So what would happen is since in a person like me and you who do not have the disease, our endonuclease will detect the damaged nucleotide and they will go and release the damaged nucleotide. And then what would happen is after you release the damaged nucleotide, DNA polymerase and DNA ligase will go and fill the part up and reseal the gap. They will reseal the gap. So if I have to tell you this, let's go on a blank page. Let's make a note. Okay, let's make a small note over here. Okay, the small note over here. The small note over here is this is just for your quick revision and recapitulation. First of all, you have name of the repair. You have name. Okay, then you have enzymes, and then you have disease. Then you have. Okay. Does anyone have a question? Does anyone have a question? If you have the question, please use the chat box. Okay, good. Name, enzyme, and the disease. First one, the first disease, the first DNA repair that we talked about was nucleotide excision repair. Nucleotide excision repair. The enzymes that we basically talked about was, first of all, if there's a damaged DNA, the enzyme that will come is endonuclease. Then you have DNA polymerase and DNA ligase. These are the three enzymes that are required to heal or repair the structure of the single-stranded DNA damaged. And the most common disease over here, if this is not working properly, then what would happen is the most common disease that will happen is xeroderma pigmentosa. Xeroderma pigmentosa. What happens is that in xeroderma pigmentosa, you have the DNA excision repair, which cannot repair the DNA pyrimidine dimers, which are caused by ultraviolet exposure. So ultraviolet exposure causes damage to the nucleotide, and we do not, and the patients, they do not have proper proper enzymes to go and get rid of those damaged nucleotides. So that's that. So patients usually have dry skin, light sensitivity or photosensitivity, and they're highly predisposed to skin cancer. Xeroderma pigmentosa occurs due to a defect of nucleotide excision repair, which is a single standard repair. This piece of knowledge is extremely high yield. This is very, very high yield. Okay. And the next type of question they'll ask you is which enzymes are not working in nucleotide excision repair, and the enzymes are these three enzymes. That's also very, very high yield. So if you have to do a quick revision and recapitulation, if you have a note like this, you can just glance through it once and you can have your revision done. Next one. Next nucleotide, next DNA repair, which you want to talk about, is base excision repair. Base excision repair, what happens is you have base specific glycosylase, which removes altered bases and creates an AP site. AP site is basically an apurinic or apyrimidinic site. AP site meaning that over there you do not have any purines and pyrimidines. So what the glycosylase does is glycosylase, they go and they remove the bases which has been damaged. Okay, they go and they remove the bases which has been damaged. After that, the enzyme AP endonuclease, one or more nucleotides are removed. Then AP endonuclease will go and remove the nucleotides by cleaving their 5' end. Okay, and then DNA lyase, not ligase, DNA lyase will go and cleave the 3' end. So endonuclease will go and cleave the 5' end, and lyase will go and cleave the 3' end. And then you have DNA polymerase and DNA ligase, which will go and seal this. This base excision repair, what this does is, in a process such as spontaneous deamination. Do you remember how we talked about deaminations yesterday? Do you guys remember how we talked about deaminations yesterday? Yes or no? And lipo and all those. Good Christmas, right? Then what else? And taco, right? And what was another one? Sorry, me taco. My apologies. Not me, not M taco. Me taco. What was the first one? U cycle, exactly. So if we have these types of spontaneous deaminations, is there a possibility that these type of spontaneous deaminations, can this predispose the patients to develop carcinomas? Because once there is spontaneous deaminations like this, then there's a high possibility that there could be unregulated and unproliferated growth of the tissues, and this can result in carcinoma. So we have heard a lot about the fact how in spontaneous deamination and toxic deaminations, these are the processes that is required or that happens in order to cause a carcinoma. So can we say that people who develop carcinomas, okay, for example, have you guys heard of the fact that you have a smoker who has been smoking for 40, 50 years who does not develop carcinoma, but then you have a smoker who smokes for 1 or 2 years and they develop with lung cancer? Yes or no? Have you guys ever heard this? Yes. Okay, so basically in a smoker who has been smoking for 40 to 50 years and they do not develop lung cancer, that is because they have excellent base excision repair mechanism. Are we clear? They have excellent base excision repair mechanism. In a smoker who has been smoking for 1 or 2 years, that toxic deamination, since they have a very weak base excision repair mechanism, they have a high predisposition to cancer. How interesting is this piece of information? Next time when your friends ask you, we know that we have a lot of friends who asks us medical question, and let's say someone asks you, why do you think people get lung cancer when they smoke for 1 or 2 years, but then you have people who do not get lung cancer when they smoke for 40 to 50 years? And now you have a solid answer. This is the answer you can provide to them, and you can tell them that there is a process called base excision repair, which repairs DNA damage. But in people who develop lung cancer in 1 or 2 years, they have a very poor or they have a very bad base excision repair mechanism. Okay, so once again, what are the enzymes that I've talked about? So these are, for example, okay, So this is for your quick revision. So you have base excision repair. You have base excision repair. The enzyme that we have talked about is gel, please. Gel, please. Okay. Gel, G-E-L. G for glycosylase, E for endonuclease, L for lyase, P for polymerase, L for ligase, and then you have E-A-S-E, which is not high yield, but these 5 enzymes are extremely high yield. Over here, endonucleases, they work at the 5', and lyase, they work at the 3'. That is also another question. Which works at the 5'? Which enzyme works at the 5'? That's endonuclease. Which enzyme works at the 3'? That's lyase. Okay, that's that. Then over here, what type of diseases will occur? The diseases are usually spontaneous deaminations, spontaneous or toxic deamination. Are we clear about this? Are we clear about this, guys? Okay. Question, yes. Well, what is your question? What is your question? What is your question, please? Yes, smoking can cause toxic deamination. What's the difference exactly between nucleotide and base excision? What was your question? What is the difference exactly between? Can you please unmute yourself, Dr. Karbasi, and ask me the question? Unmute yourself and ask me the question, please.
Yes. So like, if I would like say the difference between the nucleotide and base, so does it mean like a nucleotide like just repair like, for example, guanine or adenine, and the base like repair the whole codon, or like how I have to understand that?
Okay. Okay. So we are still confused about nucleotide and bases. Okay. Okay. Nucleoside is a base with sugar. Okay. Nucleotide is base plus sugar plus a bond, phosphodiesterase bond. Okay, 3', 5' phosphodiesterase bond. Now your question was, what was your question?
Like nucleotide and base excision repair, like just like nucleotide, I just check.
And base excision repair is basically, base excision repair is they will remove the altered bases, and nucleotide is also the same thing. They will remove the damaged bases, the same thing. Okay, basically, if this part is damaged, then they will remove it. That's exactly what they're saying. If it's a nucleotide damage, then in the nucleotide, usually the sugar, even if it's damaged, it's not a big deal. If the bonds are damaged, that is sort of a big deal. But the biggest problem is if there's a damage to any of the bases. So in a nucleotide excision repair, if there is a damaged base which they cannot get rid of, then it's called a nucleotide excision repair. If you have a base excision repair problem, then base specific glycosylase, meaning that there are specific glycosylase gene. For example, you have, let's say you have GAA. There's a specific enzyme only for this G. There's a specific enzyme only for this A, meaning that there's a specific glycosylase which will work only for this G, specific glycosylase which will only work for this A. So if there's a defect in that one, then this is called a base excision repair. But at the same time, once again, that base is this base over here. Okay, they're talking about the same thing. They're talking about the same thing. Okay.
So it's more specific to base excision repair. So it's like more for something specific.
Right.
Okay.
Specific to every nucleotide. Every nucleotide has their own specific glycosylase. Okay.
Okay. So like a nucleotide excision repair, like It's like general, like it starts to begin to
Right.
repair if it can, and then the base excision repairs to come. Okay,
Right. Okay.
thank you.
You're welcome. Okay. Any other question? Okay, so that's that. Okay. To be honest, it's good that you guys are trying to clear out your conceptions. But if we try to go into very deep genetics, then we'll be in deep problem, because genetics is very, very deep. Because to be honest, if we try to understand why base excision repair is not working and this and that, then we would have a very long lecture. But the purpose of our lectures is to understand the text in first aid, and I'm really, really glad you guys are asking me the questions. So please don't get me wrong. Please ask me as many questions as possible. Okay, but please try to keep in mind that there's absolutely not 100% we can learn about genetics, because genetics is extremely complex, and most of the things are beyond the scope of step one. Okay, so that's that. But these notes, these are exactly what you will need to do good in the step one exam, because this is how the questions are asked. Okay, does anyone have any question before we move on to the next one, please? Repeat the steps of base excision repair. Base excision repair is nothing but the fact that when you have a damaged or altered base, glycosylase will go glycosylase and remove the altered base. That will create an apurinic site over there. Then you will have two enzymes. First of all, endonuclease will remove the 5' and lyase will remove the 3' end. So basically, they want to make sure that the toxic deaminated portion of the base is removed entirely. So there is no chance for development of carcinoma. Then after removal of those damaged bases, the enzyme polymerase and ligase will go and fill the gap and seal the gap. That's it, nothing else. The only thing that you have to remember from base excision repairs are the names of the enzymes, that is glycosylase, endonuclease, lyase, polymerase, and ligase. You would have to know which enzyme works at which position. Endonuclease works at 5, lyase works at 3, and then you would have to know which diseases will occur if this is not working properly. And the diseases are spontaneous and toxic deaminations. That's that. Are we clear? Are we clear about this? Are we clear about this? Okay. Next one is mismatch repair. Mismatch repair, what happens is, right now, instead of damaged nucleotide or a damaged base, you have a mismatched nucleotide. The nucleotides are not matched properly. Because do you remember how we told you that every nucleotide has to match in order to code for the specific amino acid? And if there is mismatching of a nucleotide, then this can result in an entirely new different protein, or even in a carcinogenic protein, for example, carcinoma, right? So you have mismatched nucleotides, which are formed in the newly synthesized strands. And this mismatched nucleotides, they are removed by the mismatch repair genes, and this gap is filled and resealed. If you have any defect in this sort of mismatch repair mismatch gene repair, then you have a very important disease, which is known as Lynch syndrome, or hereditary non-polyposis colorectal carcinoma. So once again, over here, if we have to complete our table, last one is mismatch repair. Mismatch repair. There is no specific enzyme only for mismatch repair, except the fact that we can guess that the enzymes are endonuclease, ligase, and polymerase, because once again, they're talking about removal, filling, and resealing. So it could be the same enzyme. So we're going to keep this hollow. We're not going to fill this up with anything, since first aid did not mention any specific enzymes, but they did mention a disease, that is hereditary non-polyposis colorectal carcinoma, or Lynch syndrome. Next time you want to do a quick review of single-stranded DNA repair, this is all the knowledge or all the information you need over here. This is all the information you need over here. From here, these enzymes are extremely important, the names of these enzymes. Okay, and the diseases, of course. So if you have to test yourself, let's say you want to test yourself whether you remember this or not, cover this portion with your hand, ask yourself that what type of DNA repair mechanism is absent in Xeroderma pigmentosa, and try to remember that it's nucleotide, spontaneous demyelination is base, hereditary non-polyposis coli is mismatch repair. Okay, are we clear about this? Yes, okay. So what type of, can we do a quick revision of the single stranded ones? Can I give you guys 1 minute? I'll give you guys 1 minute before I move on to the double-stranded DNA damage. I'll give you guys 1 minute. Read this. I will ask you questions only if you have finished this, then we can move forward. Okay. Next one is please repeat mismatch. Mismatch is basically the nucleotides which are mismatched for some reason. They have to be removed in order for the proper amino acid to form. So when there's a mismatch in a nucleotide, there are enzymes which removes the mismatched genes, and this removal of the mismatched genes are then later filled up. The gaps are filled up and resealed. This is the mechanism of the mismatch repair. If the mismatch repair, if they do not occur properly, then this mismatching can result in the formation of Lynch syndrome or hereditary non-polyposis coli syndrome. That's that. Okay. Does anyone remember the 3-2-1 rule of hereditary non-polyposis coli? 3-2-1 rule of hereditary non-polyposis coli. Who remembers the 3-2-1 rule? Talked about this so many times. What is the 3-2-1 rule? 3-2-1 rule. How can you identify hereditary non-polyposis coli? Via the 3-2-1 rule. 3 generations, 2 affected individuals, right? And 1 over what? And what? 1 person less than 50. Very good. Okay. So, okay, very good. So there has to be, very good. There has to be 3 generations of, for example, the grandfather, father, and the son, all 3 of them need to have the diseases or need to have the gene. 2 of them, for example, the father and grandfather can have the disease, and 1 person, that is, any one of the affected individual could be less than 50-year-old. That is the onset of the disease. This is the 3-2-1 rule of diagnosing Lynch syndrome. Okay. Okay. Are you guys done about learning about the single strand layers? Yes or no? Okay. Okay. Xeroderma pigmentosa. What type of nucleotide excision repair is not working over here? Xeroderma pigmentosa. Okay. What type of excision repair is not working in spontaneous or toxic demyelination? Okay. Okay. What type of enzymes are required for base excision repair? Okay, good. What type of are required for nucleotide excision repair? Okay. What type of repair mechanism is absent in hereditary non-polyposis coli? Okay, good. Next one. Okay, very good. Very good. Okay, next one. Next one that we want to talk about. So we talked about the single-stranded DNA repairs. Now we will talk about the double-stranded DNA repairs. Okay, double-stranded DNA repairs are very easy to understand. Basically, double-stranded DNA repairs are, you have 2 types, that is non-homologous and homologous recombination. Non-homologous and joining, non-homologous and joining repair. Basically, what happens is this brings together the 2 ends of the DNA fragments to repair the double-stranded breaks. For example, if you have a break like this, and then you have a break like this, right? In this type of mechanism, what they will do is they will bring they will bring the two ends together and repair the DNA. That's that. As simple as that. That is, they will bring the two ends of the DNA fragments to repair the double-stranded break. This type of mechanism is known as non-homologous end joining. And the fact that non-homologous end joining is absent in ataxia telangiectasia. This is an immunodeficiency disorder. Have you guys heard of ataxia telangiectasia? Yes or no? Yes. Okay. So that's that. Okay. So ataxia telangiectasia is a very high yield. It's a immunodeficiency disorder, which we will talk about, which we'll talk about very soon. Okay. And this type of repair mechanism, this occurs in this. This is known as non-homologous end joining, and this is defective in ataxia. Next one is homologous recombination, which requires two homologous DNA duplexes. A strand from a damaged double-stranded DNA is repaired using a complementary strand from intact homologous double-stranded DNA as a template. So what happens is, okay, so what happens is, let's say that this is the DNA, okay, and this is the part, one strand of the DNA is damaged. Okay, so for example, one strand is damaged and another strand is okay. So what they will do is, in homologous recombination, instead of joining, they will go and they will form a complementary strand, which is identical to this part, and they will bind that to over here. Okay, so this is known as homologous recombination. The fact that they bound a complementary strand to this purple one, and then they added this one, this is known as homologous recombination. This process is defective in breast carcinoma with BRCA1 mutation and in Fanconi's anemia. This restores the duplexes accurately without loss of nucleotides. There is no loss of nucleotides over here. Okay, so if we have to make another table for this, the easiest way you can make a table for your quick revision, because only the names and the diseases are important over here. There's no specific enzyme. Okay, so you have your name and then you have your disease. The first excision repair of double-stranded is non-homologous end joining, and the disease is ataxia. Right? Okay. Over here, there is a possibility that some amount of DNA could be lost, but still it's not a big deal. And the next one is homologous recombination. Homologous recombination. And the diseases are breast carcinoma, ovarian carcinoma, and Fanconi's anemia. Breast, ovarian, and Fanconi's anemia. Over here, there is absolutely no loss of DNA. Absolutely no loss of DNA. Okay. Is anyone confident enough to answer all the questions without revision from the single and double-stranded DNA repair, for the purpose of only the questions which would be asked in you? Okay, no other deeper question, just the main things which will be asked at the U. Is anyone confident to answer the question? If you are, please let me know. Every one of you should be confident. Every one of you should be confident, okay? First of all, if you have a nucleotide excision repair, what type of disease will happen? What type of disease will happen in a nucleotide excision repair? Very good. If you have a base repair, what type of disease will happen? Phase of cell cycle where occurs is important. Phase of cell cycle is S phase. We all know that in S phase, we have everything that is either repaired, condensed, released, and everything happens in S phase. It was a very basic piece of information. At an S phase, you have everything happening, repair, and then you have the release, unwinding, right? So that's that S phase. This is very commonly asked because it's very well known that in S phase, which is the synthetic phase. Okay, that's that. Next one is what type of disease will occur in mismatch repair? Mismatch repair. Okay. Next one. This one is, what type of disease will occur in non-homologous end joining? Non-homologous end joining. Okay. What type of disease will occur in homologous combination? Okay. 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 DNA repair mechanisms. Okay. Let me see who was the physician who asked about the phases. Let me see. Okay. Dr. Dahlia. Dr. Dahlia, thank you so much for bringing that to our attention. First of all, we might not have put enough emphasis on the phases, but Dr. Dahlia was kind enough to mention this. You have everything which can occur in S phase, and nucleotide excision can occur in G1 phase for the nucleotide excision repair, but also to some extent nucleotide excision repair also occurs in S phase. So if you want to remember this, just try to remember that G1 and S phase are important for repairs, but this is not a very high yield, especially for the fact that we know that most of them will occur in the G1 and S phase, so that's that. But S phase is more common. S phase is more common. Okay, so thank you, Dr. Dahlia, for bringing that to our attention. Having said that, have we understood everything about the mutations? Yes or no? I mean the DNA repairs. Okay. All right. Next time we receive a question on the DNA repair mechanisms, can we be confident that we can answer them properly? Okay. Okay, good. Okay, let's move on to the next one. Do you guys want to take a 5-minute break before we move on to the next one? 5-minute break? Not a 10-15-minute break. I'll give you guys a 10-15-minute break. Let's just take a 5-minute break so that we can clear our head, because we will be learning completely new different things from now. Yes or no? Okay. Okay, let's just take 5 minutes. Okay, is everyone back from their break? Can we do a quick revision of today's topic before we move forward? We'll make sure you guys have learned anything. Okay, so we'll just take only 1 minute. I just want to make sure you guys have learned all the things or not. Okay, we have only discussed 3 pages. First of all, if we have a mutation and there is no change in the amino acid, what type of mutation is this? So very good. If we have a mutation where there's an early onset of stop of the mutation, I mean, of stop of the amino acid synthesis, what type of mutation is? It's an absolute mutation. If you have a mutation where there is substitution and the entire amino acid is changed, what type of mutation is this? Okay. If you have a mutation where the entire shift of the mutation has been changed and it's not divisible by 3, what type of mutation? Okay. If you have a mutation where there's retention of introns, what type of mutation is that? Okay. What type of mutation is sickle disease? Sickle cell disease, what type of mutation is sickle cell disease? Very good. What type of mutation is Tay-Sachs disease? What type of mutation is Smith syndrome and beta thalassemia? Okay, if we have low glucose and high lactose, what would happen to the lac operon? If we have high glucose and low lactose, what would happen to the lac operon? Okay, low glucose, create the lac operon. Can you guys hear my voice? The voice breaking down. What happened to the lac operon if there's low glucose? Voice. Okay. Okay, how about now? Is the voice better now? Okay. Um, okay, so high glucose and low lactose, what would happen to the lac operon? Okay, high glucose and some lactose are available. What would happen to the lac operon? Okay, if you have a nucleotide excision repair defect, what disease will happen? If you have a base excision repair, what disease will happen? Base excision repair damage. Okay, if you have a mismatch repair defect, what disease will take place? Commonly. Okay, if you have non-homologous end joining repair mechanism that is defective, what disease will take place? If you have a homologous recombination damage, what are the diseases that can take place? Okay. Okay. So these are the things. Okay. So very good. Thank you so much for retaining these informations. So that's all. Okay. Are we confident about this? Can we move forward? Okay. Very good. Okay. Let's move forward. Okay. 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 Now, The next thing that we're going to talk about is functional organization of a eukaryotic gene. The next thing that we're going to talk about is functional organization of a eukaryotic gene. Okay, this is very easy to understand. Basically, what the functional organization is, you have the DNA. DNA is transcribed to form pre-mRNA. Then we splice out the introns to form matured mRNA. Then they undergo the process of translation, and then you have the formation of the protein structure. So step by step. So what's happening over here is that instead of drawing it in a blank page, okay, I would just want to use this diagram over here. So if this is the DNA, then this is the 5' end. This is the 3' end of the DNA, right? In the 5' end of the DNA, do you remember how when we studied the lac operon, we studied how the DNA in the E. coli, they have the promoter region, then the operator region, and then the genes, right? So the same thing happens in the human DNA. Basically, at the beginning, you have this segment. This is the silencer. Then you have the promoter. In humans, the promoters are known as CAAT box or CAT box and TATA box or TATA box. The fact that the promoters are known as CAT box and TATA box, this is extremely high yield, because next time you receive a question about CAT box and TATA box, whenever they mention CAT and TATA, try to think that they are talking about some problem or something regarding the DNA, the DNA promoters, DNA promoters. Okay. Then what happens is, if you activate the promoter, then you have the onset of the DNA, of the mRNA from the DNA. So in order to do that, you have this portion, right? So this is the start codon. Basically, this is AUG, right? And then the mRNA is made from over here, and this premature mRNA, okay, this premature mRNA, this consists of exons and introns. Basically, the introns are the ones which are not important, so they have to be removed by splicing. The introns, they have to be removed by splicing. And after you remove the introns, this gray portions, you have only the exons. These exons are the ones which are important. And then you have the final mRNA. The final mRNA, they consist of the exons, they have a cap, and they have a tail. We'll talk about this in 1 second. This is just a brief description of what's going on over here. And then from over here, from over there, you have the formation of the protein. You have the formation of the protein over here. Okay, so that's that. So this is just a brief description. Don't worry about this. We'll break this down into little details, and we'll talk about what's happening. First of all, let's talk about the regulators of gene expression. Well, what are the regulators of gene expression? First, you have the promoters, you have the enhancers, and then you have the silencers. The promoters are basically, these are the site. So these are the promoters, which has the CAT box and the TATA box. This is the site where RNA polymerase 2. RNA polymerase 2 is an enzyme. Okay, they will go and work over there to synthesize the mRNA. RNA polymerase is responsible for the synthesis of mRNA. Okay, so what this will do is, so what the RNA polymerase will do is that if they want to synthesize the mRNA, they will go to the promoter part, that is the CAT box and the TATA box, and they will go and stimulate this portion. So next time, if you receive a question that where does the RNA polymerase 2, where does it go in order to promote the mRNA synthesis? And your answer should be the promoter sequence, that is the CAT box and the TATA box. Because if you stimulate the promoter, then you can have the synthesis of the mRNA from the DNA. So this is the site where RNA polymerase 2 and multiple other transcription factors. You know, like how we have a lot of transcription factors. Do you guys remember how we talked a lot about growth factors, EGFR, FGF, then VEGF, right? Those are all factors. Am I correct? Yes or no? Do you guys remember the factors? Transcription factors? Yes. When we have excess of those factors, do you remember how we told you that those factors, when they will go, they will cause extreme protein synthesis? They will increase the protein synthesis. The fact that they can increase the protein synthesis, those growth factors, they go and they directly bind to the promoter sequence. So this is the information which you need. Next time when you receive a question, where does RNA polymerase 2 and the transcription growth factors, where do they go to stimulate the mRNA synthesis? And your answer should be the promoter region, that is the cat box and the Tata box. They bind to the DNA from the gene locus at the AT-rich upstream sequence with Tata box and the cat box. That's what it is. The promoter mutation commonly results in dramatic decrease in genetic transcription. This makes sense, obviously, because if you have any mutation happening over here, then there is no promotion, and as a result, this part is not activated at all. Okay, so if there's any mutation in the promoter region, then you can have genetic transcription. Okay, now let me ask you, these things that you are studying right now, for example, a lot of us will become physicians and a lot of us might become researchers. If you are a researcher, this piece of information, the fact that promoter regions are required to be activated in order to produce DNA or I mean, an amino acid, is this important or not important? For example, right now in the coronavirus pandemic, we have the formations of the vaccines, right? We have the formations of the vaccines. All these researchers who are working actively, they are also physicians. They're not clinical physicians, but they're research physicians, right? So this piece of information, if you have any student who is wondering, why do I have to learn these things? Okay, it's important because you have to apply these knowledge if you have to come up with research solutions. So that's that. Next one is, now you are a researcher. Now what you want to do is, after you have promoted the DNA transcription, the next thing what you want to do is you want to increase the regulation of protein that you want to create. For example, let's say you want to create a certain protein in your research lab. Now you have promoted it. Now you want to increase the synthesis. What you will do is you will go and stimulate the enhancer portion. When you go and stimulate the enhancer portion, that is this one over here, okay? If you stimulate the enhancer, this will increase the protein synthesis, increasing the expression of a gene on the same chromosome. So you can increase the expression of a gene on the same chromosomes. So you can increase the protein synthesis. Enhancers and silencers, they may be located close to or far or even within the gene whose expression they regulate. So they can be presented anywhere close to any gene over here. So let's say there's a particular gene which you want to increase. There's a particular gene whose protein you are trying to target, and you want to increase the protein from that gene. So what you want to do is, in order to increase the protein synthesis from that gene, you want to go and target that enhancer portion of the gene. So you can go and regulate the protein synthesis accordingly. And in the same way, how you enhance the protein synthesis and the enhancer segment, you can also go and silence and decrease the expression of the gene on that same chromosome. So what that will do is, right now, let's say instead of you trying to make more proteins, you want to make less proteins. So what you do is you activate the silencers. When you activate the silencers, they will decrease the expression of the genes. What do you have to know about this in order to do well in the exam? That is, you have to have the knowledge that is the promoter regions are known as cat box and Tata box. When you activate the promoters, there's excessive genetic transcription. When you want to regulate a protein and you want to express a single gene on the chromosome, the thing you want to activate is enhancer. And the opposite is, when you want to decrease the expression, the thing you want to activate is a silencer. Okay. Are we clear about this? Yes or no? Are we clear? Very easy to understand. Okay. Very easy to understand. Next one is, so what happens is, after this one, okay, before the formation of a premature mRNA, you have a structure which is called heterogeneous nuclear RNA. Let's talk about this over here. You have a structure which is called the initial transcript. Initial transcripts are basically, they're the first transcripts that are formed from the DNA. So you have the DNA 5 prime, 3 prime, and the first structure that is formed is HNRNA, heterogeneous nucleus RNA. Heterogeneous nucleus RNA is modified and then it becomes premature mRNA. How does this modification occurs? This modification is very important, and you have to remember the steps. What happens is, what happens is over here. Okay, just can you guys just give me 1 second, please? Just have a very important phone call. Just 1 second. Okay, give me 1 second. Okay, my apologies. I'm sorry. Okay. So let's get back to the lecture. So what we were talking about, so let's say that this is the DNA. The first transcript that is formed is hnRNA, which is known as heterogeneous nuclease RNA. Okay, so let's talk about something else. Okay, let's say that we have a newborn baby. Okay, let's say we have a newborn baby, and after the baby is born, do you know how parents, they usually try to dress the baby up in a cute way, right? Try to dress up their hair, cut their nails, make them wear new clothes, and so far and so forth, because they want to make sure that the baby is prepared to go out to the world and everyone can go and see the baby. Yes or no? Yes or no? Are you guys still with me? Are you guys still there? Okay. So what happens when we have a new baby? What happens when we have a new baby? What happens is the parents, what they try to do is they try to dress the baby up. Okay, so how do they dress the baby up? First of all, let's say that if this is the baby, okay, okay, if this is the baby, the parents, what they do is, first of all, the parents, they make the baby wear a cap. Okay, they make the baby wear a cap. Okay, the baby wears a cap because they want to make sure that the baby looks cool, that then the baby looks okay, and also because they want to make sure that the baby doesn't have anything which can go wrong with the baby's hair. Okay, so the parents make the baby wear a cap. After that, the parents, what they do is they cut the baby's nails. They want to make sure that the baby, they do not have sharp nails. Okay, so cutting nails. So they cut the nails. Okay, so nails, are nails important or not important? Yes or no? Nails, are they important to us, or are they not important to us? Can we cut the nails? Can we cut our nails? Nails are basically, long nails are not important. So let's say that cutting nails, are the same as nails can be the synonym for introns. Introns. For example, how we do not need introns. At the same time, we also do not need long nails. So we can get rid of long nails and we can also get rid of introns. So cutting nails, can we say that cutting nails are almost the same as splicing introns? Splicing introns. Okay. Yes or no? Okay, good. Another thing the parents do, okay, the parents, well, what they do is they are a bit humorous. So what they do is they make the baby wear dinosaur cloth where they have a tail. Okay, you know, like how those cute baby dinosaur clothes which the babies wear. So that type of a cloth which the parents make the babies wear, they have a tail. So this is tailing. Okay, so the fact that the parents made the baby wear a cap. This, you have to remember that in order to produce a premature mRNA from a heterogeneous nuclear RNA, the first process is capping of the 5' end. You have to cap the 5' end by a 7-methyl guanosine cap. In order to produce a premature mRNA from a heterogeneous nucleus RNA, the first process is capping by a 7-methyl guanosine cap, 7-methyl guanosine cap. The next one is splicing of introns. Then what you do is heterogeneous nucleus RNA, they contain a lot of introns. And we know that introns are not important. And the next thing which the parents did was cut the nails of the baby. So splicing of introns are the next step. So the next step is splicing of introns. And then the last step is the parents, they made the baby wear a cloth with a dinosaur tail. So tailing. Tailing is for polyadenylation of 3' end. At the 3' end, we add around 200 adenines. 200 adenines. So in order to make mRNA, they have to have a cap at the 5' end. They have to have no intron. And at the 3' end, they have to have 200 adenines, polyadenylation at the 3' end. These steps are extremely important for your exam. That's why I made this mnemonic over here with the baby, so that you guys can never forget that in order to produce an mRNA from heterogeneous nucleus RNA, you have to go through this process. Are we clear about this? Yes or no? Do we understand this? Did we understand this? Okay. So basically, RNA processing is initial transcript is called heterogeneous nucleus RNA. It is then modified and this becomes mRNA. What are the process of modification is capping of 5' end by a 7-methyl guanosine cap. Then polyadenylation of 3' end, polyadenylation, and splicing of introns. That's that. Okay, so hopefully you guys will not forget this. This is extremely high yield. The capped, tailed, and spliced transcript is now called an mRNA. Okay, next one is mRNA is transported out of the nucleus to be translated in cytosol. And then the mRNA quality control occurs at cytoplasmic processing bodies called P bodies. This is extremely high yield. So what happens is after the mRNA is formed, it is transported from the nucleus to the cytoplasm. In the cytoplasm, the quality control, meaning the regulation of mRNA, is occurring at a very important organelle, which the USMLE will test you. That is cytoplasmic processing bodies, P bodies. This is very, very high yield. Okay, what would happen over there? Over there, they contain exonucleases, which will decap the enzymes and microRNAs, and mRNAs may be degraded or stored for future translation. So basically for regulation. So what happens next time when they ask you that after the mRNA, this is an NBME question, guys, that what happens after the mRNA comes out of the nucleus? After the mRNA comes out of the nucleus, it goes to the cytoplasm. In the cytoplasm, it enters the cytoplasmic P bodies. In the cytoplasmic P bodies, they are usually stored for future translation, or they can be decapped and microRNAs can be removed. That's that. And polymerase, okay, this is basically the enzyme that is required for polyadenylation. That this is not very high yield, and AAA is the polyadenylation signal, not high yield once again. Okay, that's that. Is everyone clear about RNA processing? My apologies, I'm sorry. Is everyone clear about RNA processing in eukaryotes? Everyone? Okay. Cytoplasmic P bodies. Cytoplasmic P bodies will store the mRNAs for future translation, or they will decap the mRNA or remove the microRNA so that they are prepared to undergo translations. They are used for the regulation of mRNA function. mRNA processing occurs in the nucleus. Yes. Okay. Any other question? What is your question?
Dr. Yesterday we spoke about CPG island, right?
Mhm.
It's written there that they work on
Where?
the repressor gene, but gene promoter CPG island typically repressor gene. How is it possible that promoter and repressor gene are together? Because
Wait, one
now we
second.
know that promoter gene.
One second. Where are we talking about this?
We talk about it in the first page of our topic.
No. I remember CPG islands, but where are we talking about? What are we talking about this one?
Methylating.
Over here?
No, above the gene. Enhancer,
Where?
silencer, and promoter, doctor.
Silencer and promoters over here.
Yes, the island where it's working exactly, where it's localized.
You mean where is the location of the CPG islands?
Yes.
The location of the CPG island is in the promoter region. So it's in the promoter region. Let's go back to the first page. Let's see where the confusion is. Okay, no problem. Let me clear the confusion. Just give me 1 second. Okay. 1 second. I don't know why this takes some time to load. Okay. One second. Okay. All right. Okay, Dr. Mohammed, are you there with me?
Yes.
Okay. So over here, it says that DNA methylation within gene promoter CPG islands, typically they repress the genes. So you have CPG islands. These are within the gene promoter region. If you activate the CPG islands within the gene promoters, not activate, my apologies. If you methylate the CPG islands and the gene promoter, then you can cause DNA mute. I mean, you can mute the DNA. And at the same time, over here, first of all, you have the enhancer, then the promoter, then the augmenters, and then you have the regular genes. If you activate the enhancer, this will go in the same way. If you activate the promoters, this will go in the same way. If you inactivate the enhancers, meaning that you activate the silencers, this will not go the same way. Or if you do not activate the silencers, but you want to methylate the gene promoter region, that is the CPG islands in the promoter region, then this will also inactivate. So the process is the same. The mechanisms are just a little bit different. Are we clear about this?
Of course. Thank you very much, doctor.
Okay, no problem. That was a good question. I'm glad to clear the confusion. Any other confusion? Any other confusion? Do we have any other confusion, guys? What is microRNA? We haven't talked about microRNA yet. We will talk about this right now. Any other question except that? Okay. If I hear you guys responding less, can I assume that you guys are tired? Or is that what it is? Are we tired right now? Is that the problem? Okay. Okay. So do you guys want to take a break or what? Yes or no? Before we move on here. Before we move on to this page, do we want a break? Or do we want to keep moving on? Okay. How long do you guys want to take the break for? 10 to 15 minutes? Okay. So let's take the break for 10 to 15 minutes and let's come back. It's 11:25. Let's come back at 11:40. Okay. Okay, is everyone back from the break? Is everyone back from their break? Okay. Shall we begin with the lecture and finish with the genetics? Okay, good. Okay. So let's begin with the lecture. Okay. So right now we are going to talk about RNA polymerases. We'll talk about RNA polymerases. Just as how DNA, do we remember the function of DNA polymerase 3 in DNA replication? How it was forming the leading strand and the lagging strand. And RNA polymerase, they basically work in more or less in the same way. That is, RNA polymerase will make an mRNA. Basically, they make RNA. RNA polymerase means they make an RNA from the DNA. So there are three types of RNA polymerase. There is RNA polymerase 1, there's RNA polymerase 2, and then there's RNA polymerase 3. So RNA polymerase 1 is responsible for making rRNA. RNA polymerase 2 is responsible for making mRNA, microRNA, and snRNA. Okay, and RNA polymerase 3 is responsible for making 5S RNA, TN, TR, and tRNA. Okay, the fact that RNA polymerase 1, they make ribosomal RNA or rRNA. A ribosomal RNA is the most common type, and it's present only in the nucleolus. So since RNA polymerase 1, they will make rRNA, and this is the best type of RNA, meaning that they are the most common RNA. So increase in number. So number 1 meaning most common. So RNA polymerase 1, most common. So that's how you can remember. Most common is rRNA. That's why rRNA is made by polymerase 1. Polymerase 2 is responsible for making mRNA. So we have the polymerase 2. Okay, we have polymerase. This is POL. Okay, 2 is responsible for making mRNA, right? MicroRNA and snRNA. Okay, and RNA polymerase 3, T43 and T4 tiny, or T4 tRNA. So polymerase 3 for tRNA. Okay, the fact that RNA polymerase 1 is responsible for this, polymerase 2 is responsible for these, and polymerase 3 is responsible for this is extremely high yield. You will receive a lot of questions from this. That is, which RNA polymerase forms which type of RNA. That's that. But how DNA polymerase, they have proofreading abilities. That is, they want to see if they made the proper DNA, right? But when RNA polymerase, they make the RNAs, they do not have those proofreading functions. So they make the RNA, but they cannot go and check if the RNA they made is the right one or not. So they have no proofreading functions. They can initiate chains. RNA polymerase 2 opens DNA at the promoter site. The RNA polymerase 2, they open the DNA at the promoter site, right? So what happens is you have the promoter sites over here. Okay, at this size, the RNA polymerase 2, they come and they open the DNA up, and then they perform their function. So that's that. So I want to talk about the pathology behind RNA polymerase 1, 2, and 3. The RNA polymerase 1, 2, and 3, there are certain substances which they go. So for example, if I ask you right now that if I stop the RNA polymerase 2 function, will there be proper mRNA formation? Yes or no? Yes or no? If I go and stop the RNA polymerase 2 function, will there be proper mRNA function? Okay, so if there is anything which is attacking RNA polymerase 2 and the mRNAs are not formed, what happens is, for example, the liver. Is liver a high metabolic organism? Yes or no? Yes. So liver is responsible for a lot of toxic metabolism. A lot of the toxins in the blood, drugs, everything is metabolized in the liver, more or less. So what happens is the tissues of the liver, they undergo a lot of degeneration and regeneration. So if you block RNA polymerase 2 in the liver, which is a highly metabolic organ where transcription is extremely important. So if there is damage to the liver during normal physiological metabolism, that is by drugs and toxins, the liver needs to regenerate. But if the RNA polymerase 2 in the liver is not working, can normal metabolism cause liver damage? Because the liver now cannot regenerate. That's why there is a type of toxin. This is known as alpha amanitin. Alpha amanitin. This is known as this is a toxin which is found in Amanita phalloides. Okay, Amanita phalloides. These are mushrooms. Okay, these are mushrooms. Okay, how many of us are well acquainted to the fact that potential mushrooms are hallucinogenic drugs which are abused by drug addicts to enter a state of hallucination? How many of us are well acquainted with the fact that how mushrooms are hallucinogens which are used by drug addicts to enter in a state of hallucination? Okay, so we have heard about this, right? So what happens is if you have mushrooms, okay, if you have mushrooms, death cap mushrooms, these death cap mushrooms, what they do is the death cap mushrooms, for example, instead of selling a drug addict a normal hallucinogenic mushroom, let's say if the drug addict has alpha amanitin containing Amanita phalloides, which is a type of mushroom, then they can die from liver they can die from hepatotoxicity because alpha amanitin, they attack RNA polymerase 2. If they attack RNA polymerase 2, then you do not have proper functioning of the mRNAs. And with the high amount of metabolism that occurs in the liver, there is degenerations, but there is no proper regeneration because the tissues of the liver, they cannot form. So that's why the patients, they can die from hepatotoxicity. Are we clear about this? This is a high yield question. This is a high yield question from USMLE. Amanita phalloides, which RNA does it affect? The answer is RNA polymerase 2. What is the cause of death? The cause of death is hepatotoxicity. Okay, another one is actinomycin. Actinomycin. Okay, next one. If we have a cancer, for example, if we have a carcinoma, in carcinomas, do we have unregulated and unproliferated growth of proteins? Yes or no? Okay, good. So if we stop RNA polymerase 2 from working over there, can we stop the excessive protein growth? Yes? Okay. So what happens is actinomycin is an oncological drug, it's an oncological drug which can go and block all type of RNA polymerase 1, 2, and 3. So these are actinomycin. They will go and block RNA polymerase 1, 2, and 3, and they will stop protein synthesis. And they can be used in both eukaryotic organisms, such as humans, and prokaryotic organisms. Okay. Are we clear about the pathology behind RNA polymerases? First one is amanita phalloides, which contains alpha-amanitin, which blocks RNA polymerase 2, prevents the formation of mRNA. As a result, the first organ to be damaged is the liver because of high amount of liver metabolism, which occurs over here. So there is only degeneration. And
Hello. Hello.
Okay. Do we have any questions? Okay. Do we have any questions? Actinomycin also causes hepatotoxicity? No, it does not cause hepatotoxicity. Actinomycin is only it's a drug which is responsible for blocking RNA polymerases. Okay. We will do a quick review later. I have to finish this first, so please hang on and wait. We will do a quick review when the time is right. Okay. Does anyone else have any question? Have any more question? You will get the video of the recording. So if there's anything which you think you need a revision of, okay, there's always the possibility of rewatching the videos. So you can do that. If you have any questions regarding RNA polymerases, then now is the time for you to ask. Do you have any question? So liver damage. Okay, why does actinomycin not cause hepatotoxicity? Actinomycin can cause hepatotoxicity, but the thing is, actinomycin is given in controlled amount only to stop oncological carcinomas. Oncological carcinomas, meaning not oncological carcinomas, I mean carcinomas in general, we give actinomycin to prevent RNA polymerases, but we give that in controlled amounts to make sure that there is no side effect. But if there has to be a side effect, then hepatotoxicity can happen in actinomycin. But since it's a drug, it's a pharmacological drug, and we give it in a controlled amount, that's why we do not get the side effects. Just as how every drug has a side effect, actinomycin can cause hepatotoxicity. But alpha-amanitin is a toxin. It's a toxin. It's not prescribed. The people have it by mistake or by something else, and they end up dying, or they end up passing away because of the high amount of liver toxicity which alpha-amanitin causes, because it stops RNA polymerase 2. Okay, perfect. Next one is, these are the clinical basis behind RNA polymerases. And these are the RNA polymerases in eukaryotes, that is in humans. In prokaryotes, you have only one type of RNA polymerase, which is responsible for making all types of RNA, So that's that. And in prokaryotes, the very high yield drug is rifampin. Rifampin will inhibit DNA-dependent RNA polymerase, and they will prevent new protein formation. And we know which disease we prescribe rifampin in. Rifampin is very well known for being prescribed in which disease. Does anyone have any idea? Very good. In tuberculosis. Okay, that's that. Has everyone understood RNA polymerases? Okay, good. Next one is splicing of pre-mRNA. Part of the process by which pre-mRNA, precursor or premature mRNA, is transformed into matured RNA. So what we do with splicing is basically, as we have discussed before, splicing is the removal of the introns. Alterations in snRNP, meaning small nuclear ribonucleoprotein assembly, can cause clinical diseases. Example, in spinal muscular atrophy, snRNP assembly is affected due to SMN protein. Do you guys remember we talked about spinal muscular atrophy in demyelinating disorders? Yes or no? In neurology? Yes? Good. So splicing over there, that's also a splicing defect. What happens is over there, if there is an snRNP assembly defect, then you get a decrease in SMN protein, which is a congenital degeneration of the anterior horns of the spinal cord, and the babies are floppy baby. Okay, so in neurology, there are demyelinating diseases. So if you go to neurology, then over there in the demyelinating disease, the number one disease they talked about was spinal muscular atrophy, which is a congenital demyelinating disorder of the anterior horns of the spinal cord. Over there, what happens is, since the reason of the development of spinal muscular atrophy is a splicing defect, meaning that over there you have an alteration of ribonucleoprotein assembly, right? And that protein assembly cannot occur because in splicing, we are supposed to remove the introns, but in spinal muscular atrophy, the introns are not removed. As a result, the proteins are malformed. As a result, there's decreased formation of a very high yield protein, which is SMN protein. And due to decreased activity of this protein, there's congenital degeneration of the anterior horn cells. If you do not understand this right now, no problem. When we study neurology, then you will understand it then. Or if you have already studied neurology, then there's absolutely no reason why you will not understand this, because we have mentioned this in neurology, that in spinal muscular atrophy, it's a splicing defect, because in the anterior horn cells of the spinal cord, you have degenerations of the anterior horn cells, which is a congenital degeneration. Okay, that's that. Now let's jump into the process of splicing. What are the process of splicing? First of all, what happens is, can you guys tell me what type of an mRNA this one is? What type of an mRNA is this? What type of an mRNA is this? What type of an mRNA is this one? How can you identify premature or precursor mRNA? How can you identify a precursor or premature mRNA? Very good. If it contains introns, then it's a precursor or premature mRNA. Over there, you have introns. Okay, over there, you have introns. And you also have, so what happens is, there are U1 snRNP, meaning small nuclear ribonucleoproteins. So ribonucleoproteins, what they do is they are used to make a loop of the introns. So it's easy to remove the introns altogether when there's a loop like this. Imagine if there's a loop like this and then you have a scissor, it'll be easy for you to cut the scissors, to cut the introns with the scissor at these 2 points and get rid of this whole loop. U and snRNP are a type of proteins, ribonucleoproteins. They are used to make a loop of introns. So what they do is the primary transcript, they combine with small nuclear ribonucleoproteins and other proteins to form a spliceosome. This loop is known as spliceosome. So if anyone asks you, what is a spliceosome? Spliceosome is basically nothing but, it's nothing but snRNP plus introns. Okay, snRNP plus introns. Spliceosomes are nothing but snRNPs plus introns. So when you have a spliceosome, then you can easily cleave out at the 5' site over here, and then this cleave point is then joined together by a phosphodiesterase bond, and the cleavage at this 3' site. So you cut it at the 5' and then you cut it at the 3', and then you add a 5, and then you add a phosphodiesterase bond, and the spliceosome is removed along with the introns it contains. That's that. So if I tell you, okay, now try to understand this. Okay, now try to understand this. If you have a disease where U1 snRNP is damaged, can you have removal of the introns? Yes or no? Okay, so in those diseases, can you have defective protein formations? Okay, I'm not sure about the types of disease where UNS and RNP is damaged, but next time if you receive a question in UWorld, so what UWorld will do is it will try to test whether you know that UNS and RNPs are required to make a spliceosome. So they will tell you about some disease that they have decreased, that they are targeting the U1 snRNP or the U2 snRNPs. And if you come across these type of diseases, try to understand that the main problem is retention of introns. As a result, the proteins are defective, and then you will have your answer very soon. Okay, are we clear about this? Are we clear about this? Yes or no? Okay. Good. Now, introns versus exons. What are introns? What are exons? Okay. Introns versus exons. First of all, introns and exons. Exons contain the actual genetic information, which will code for the protein. Introns do not code for the protein, but are important in genetic regulation and expression. What happens is you have different exons, which are frequently combined by alternating splicing site to produce a large number of unique proteins. Okay, look, so this is what happens. Let me break down what happens. Okay. 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 So, for example, let's say that, okay, let's say that this is a, okay, let's say that this is a part of a DNA. Okay. Okay, let's say that this is the part of the DNA, right? And over here, what you have is you have exon 1, and then it's an intron, and then it's an intron. Then you have exon 2, then another intron. Then you have exon 3, then you have another intron, and then you have exon 4. Okay, what is this structure? This structure is, I'm sure you guys know what this structure is. This structure is a pre-mRNA. Okay. My apologies. I'm sorry. My apologies. This structure is a DNA. Okay, so DNA basically it contains these introns and exons. So what happens is from the DNA, when you have the formation of the HNRNA, right? When you have the formation of the HNRNA, the HNRNA also contains a structure like this. So this could either be a DNA or this could either be an HNRNA, because you have the formation of the RNAs, which consist of introns and exons. Now, by the mechanism of splicing, that is by the use of U1 SNRNPs, we know that they form a spliceosome, right? And then we cleave the spliceosome at the 5' and then at the 3', and we get rid of them. So what happens is after you cleave it, you can form, what you will have is you will have left behind only the exons. So you have E1, E2, E3, E4. And these exons in this sequence will produce protein A, for example, protein A. Okay, now, instead of producing the exons in this way, instead of getting rid of this exon, this exon, this exon, how about you get rid of this exon first, this one second, and this one third? So you can have something like E2, E1, E4, E3. And this will, what this will do is this will produce another type of protein. This is known as protein B. Once again, you can also cut it at this one, and then this one, and then this one, and this will produce another type of protein, that is E4, E3, or E2, E3, E1. And this will produce protein C. This mechanism of splicing out the introns at different positions, this is known as alternative splicing. Are we clear about this? Are we clear about this? No, we're not clear about this. Why are we not clear about this? Unmute yourself, Dr. Mohammed, and ask me the question. Dr. Mohammed, unmute yourself, please. What is your
I
confusion?
didn't understand the alternative splicing, Doctor.
Okay, stay with me, do not leave. Okay, this is an HNRNA. Okay, over here, HNRNA, it consists of exons, introns, exons, introns, exons. In order to produce a protein, do you have to get rid of the introns, yes or no, Dr. Mohammed?
Yes.
Okay, good. So if you get rid of the introns in one way, for example, you cut this intron first, this intron second, this intron third. What you will have is you will have a protein with the mRNA exons in this sequence, E1, E2, E3, E4, and this will code for one type of protein. Okay. If you cut this
Okay.
intron and then this intron and then this intron, then this will change the exons. And as usual, the protein will change and vice versa. So this mechanism of splicing at different sites is known as alternative splicing. This is just a concept. Are we clear about this now? Yes or no?
So the change in the splicing of the
Right. Alternative splicing
intron
is the
is
mechanism
the alternative
of,
splicing.
right. Alternative splicing is the mechanism of cutting the introns at different places in order to produce exons with unique protein forming properties. Okay.
Understood. Yes, thank you.
Okay. Perfect. So that is alternative splicing. So that's what it is. So as you can see over here, that you have the hnRNA. These are the introns. These are the exons. And the first splicing was 1, 2, 4, 5, 6, and this produced one type of protein. And then they cut it at different regions. This produced another type of protein. Then they cut it at different regions again, and this produced another type of protein. This is known as alternative splicing. So alternative splicing, as a result, from one DNA, you can actually produce a lot of different types of alternative proteins depending on how you splice the proteins. Okay, basically, if you think about a piece of meat, okay, if you think about a piece of meat, you know, like how from a piece of meat you can make different types of dishes. You can make kebabs, you can make lamb chops, you can make so many other things, but the main thing is still the protein. It depends on how you cut the meat. Am I correct? It depends on how you cut the meat. You can make lamb chops, you can make lasagna, you can make once again kebabs. Okay, I really miss kebabs. It's been so long since I've had kebabs, dude. Okay, so that's that. So it depends on how you cut the meat, right? So that's what it is. So I can.
It's
No,
like coffee, doctor, like
so what?
coffee with milk. When you put it on the milk, it's latte. When the milk is after, so it's cappuccino. Like.
Right, basically that's what it is. Okay, no offense to the vegans and vegetarians in the group. Okay, my apologies. I do not, I do not like meat. I hate meat, as a matter of fact. Okay, I would like to stay away from meat. The older I get, the more I want to stay away from meat and have more veggies and vegans. Okay, so no offense to you guys. Okay, so that's that. So basically the example of the meat was, you have a piece of meat over here. You can cut it in different ways to come up with different parts of the meat. You can come up with steaks, kebabs, this and that. So that's the same way. You have the hnRNA. If you cut it in a single way, you have this type, you have this type. So this is known as alternative splicing. They can produce a variety of protein products from a single mRNA sequence. Okay. Transmembrane versus secreted Ig, tropomyosin variant in muscle, dopamine receptors in brain. These are all just the different examples of protein which can be made from a single type of a DNA and hnRNA. That's that. Okay, so introns are intervening sequences and exons are expressing. So introns will stay in the nucleus, whereas exons will exit and are expressed, right? Obviously, because exons will leave, because after you splice, then you have the mRNA, and the mRNA will leave the nucleus. And before this, hnRNA and premature mRNA will stay in the nucleus. So that's that. Are we clear about this? Alternative splicing? Yes or no? Okay. Okay. Okay, difference between premature mRNA and hnRNA. Okay, this is a bit late to be asking these questions because we have talked about this so many times, but no problems. We'll talk about it again. Difference between hnRNA and premature mRNA. Where do we talk about this? Over here. The initial transcript is called hnRNA. And after you modify the hnRNA with a 5' cap, polyadenylation and splicing, you have pre-mRNA. Okay. Any more questions? Gotcha. Okay. All right. So let's move on. Okay. Please step of splicing pre-mRNA. Steps of splicing of pre-mRNA. Once again, you have this one. Okay. You have hnRNA, which consists of exons, introns, exons, introns. Over here, you have these ball-like structures called U1 or U2, small nuclear ribonucleoproteins. What they do is they help form the introns in a loop. So they come over here and they make a loop of the introns, right? The balls, S and RB, they make a loop of the introns. This loop is known as a spliceosome. After that, you have splicing, which cuts the spliceosome at the 5' and then at the 3'. And then they have the formation of only exons. Okay, this is the mechanism of splicing. Any other question? Any other question? Okay. If you guys have any more questions, please stick around till the end of the lecture, where if you have questions, I will break it down once again. But if not, I want to move forward because I want to finish this. Let's talk a little bit about tRNAs. tRNAs, first of all, from our basic knowledge of science, basic science knowledge, I'm sure we have all studied about protein synthesis in high school if we had biology or anything else. Transfer RNA. What is the primary function of transfer RNA? tRNAs. What is the function of tRNAs? Anyone? tRNAs. What is the function? Transfer of? Okay. Okay. Basically, the transfer RNAs, they will read what is there in the mRNA, and then they will go to the cytoplasm and they will bring the amino acids in the cytoplasm to make the proteins where? In the ribosomes. Am I correct? Yes or no? Yes or no? Yes. Okay. So basically put, the DNA contains the information. After there is all this mechanism of splicing and this and that, you have the formation of the mRNA, which is the transcription. mRNA consists of informations for tRNAs, which goes to the cytoplasm and brings the amino acids to the ribosomes for the formation of the protein. Okay, so what happens is the structure of the tRNAs, they are formed from 75 to 90 nucleotides. Once again, nucleotides are base with sugar with phosphodiesterase bond. They are a secondary structure and they have a clover leaf form. There's this tRNA. The anticodon end is opposite. That is this one. Okay, that is this one. This is the anticodon, or okay, anticodon end. This is opposite to the 3' amino acyl end. Amino acyl end is this one over here. Amino acyl end is responsible for binding to the amino acids. Okay, so the amino acyl end. So what happens is all the tRNAs, both eukaryotic and prokaryotic, have CCA at 3' end. They have CCA at 3' end. And whenever you see a CC end at any end of the tRNAs, try to understand that that end is the amino acyl end. This piece of information is extremely high yield because even during your USMLE examination, if you're not sure which end is the amino acyl end, try to look for the CCA end. So CCA at 3', or they will ask you another thing, that is, what are the nucleotides at the 3' end? And the answer is CCA. Do not forget this. Okay, the way I like to think about it is CCA is charge. CCA is charge. Instead of H, you have another C. So CCA, do not forget CCA. The amino acid is covalently bound to the 3' end of the tRNA, that is this one over here, and this is a covalent bond. The CCA can carry amino acid. So this is a mnemonic from the first aid. Either you can use charge or you can use CCA, whichever you prefer. Then the tRNAs, they have a T arm, that is this arm over here. This arm consists of ribothymidine, pseudo-uridine, and cytidine. So T, W, and C. They are necessary for tRNA ribosome binding. So this is the arm which is responsible for binding to the ribosomes. Then you have the D arms. D arms consist of dihydrouridine, which is responsible for tRNA recognition by the correct amino acid tRNA synthetase. So this one is responsible for the amino acid tRNA synthetase, right? And then what happens is this D arm will allow detection of the tRNA by amino acid tRNA. So this D arm will detect another tRNA. So in order for the complete protein to form, a lot more than one tRNAs obviously will move around in the cytoplasm to bring the amino acid. So every tRNA has to be identified by another tRNA of the same protein formation. So this D arm is required to identify another tRNA by identifying amino acid tRNA synthetase. So that's what the D arm is for. And the attachment is the 5' CCA, 3' is the amino acid acceptor site. So this is the amino acid acceptor site. And the thing is, how does the tRNA know which type of amino acid they're supposed to get? The information is over here. The information is over here. That is in the anticodal loop. So once again, let me break it down into small baby steps. Okay. Give me 1 second. I know a lot of you can get a little bit confused at this point. Okay, just stay with me over here. First of all, what's happening? You have a DNA. Okay. Now DNA contains the information for forming a protein. Okay, good. So what happens is DNA will form. From DNA, you will have mRNA. mRNA, basically, it consists of all the exons, right? Because the introns were removed in the nucleus. mRNA, it leaves the nucleus, comes to the cytoplasm, goes to the ribosome. After that, the tRNA, it goes to the ribosome and it detects the mRNA by this process, and it forms the anticodon. So it forms the anticodon. After it forms the anticodon, it goes to the cytoplasm, right? It goes to the cytoplasm, looks for amino acid according to the anticodon of the mRNA, right? So it has this structure over here, this clover leaf structure. My apologies. This clover leaf structure. Okay. And this is the CCA. This is the anticodon. Anticodon, which consists of the code of which type of amino acid they're supposed to bring from the cytoplasm. Okay. And this one is the T arm. T arm. The T arm will be responsible for binding to the ribosome. So it consists of 3 types of protein, which is known as TWC. TWC is ribothymidine, pseudo-uridine, and cytidine. So that's that. And over there, this arm is known as D arm. D arm is known as the arm that is responsible for binding to another type of tRNA in the ribosome so that the protein can be formed. So once it finds the protein, according to this one, the protein is bound to the amino acid tRNA, that is the CCAN, and then it goes to the ribosome, and then it forms the protein. This is the mechanism of protein synthesis. Okay. So what happens is over here, we have just got done talking about the structure. Okay. You will receive a lot of questions from over here. Basically, the questions are according to the fact that whether you know that the CCA
is the amino acid tRNA end. The T arm is responsible for binding to the ribosomes. D arm is responsible for binding to another tRNA, and the anticodon loop contains the codes which are taken from the mRNA for the type of amino acid they're supposed to bring from the cytoplasm to the ribosomes. Have you guys understood this? Any questions? Any question? Okay, I suppose there is no question. Any more question, guys? Okay, good. So what happens is after this, the next step is known as charging. Charging is basically amino acid tRNA synthetase. Okay, amino acid tRNA synthetase will use ATP, that is one unique enzyme per amino acid, and binding of charged tRNA to the codon are responsible for the accuracy of amino acid selection. So basically, charging is the binding of the amino acid. Amino acid tRNA synthetase will match an amino acid to the tRNA by scrutinizing the amino acid before and after it binds with the tRNA. So amino acid tRNA synthetase will see whether the amino acid that has been bound to the tRNA is the correct amino acid or not. Okay, this bounding is known as charging. If by any chance a mischarged tRNA reads the usual codon, my apologies, a mischarged tRNA will read the usual code, but insert the wrong amino acid. For example, if the tRNA is not charged properly, they can actually bind to a different type of amino acid, which is a lot different from the code that is over here. So this is known as a mischarged tRNA. You will not receive a lot of questions from this paragraph. This paragraph is not very high yield. Okay, this paragraph is not very high yield. But have we understood the structure of the tRNA? The structure of the tRNA is exceptionally high yield. What's happening at the T arm? What's happening at the D arm? Okay. What is the T arm responsible for? What is the T arm responsible for? Binding to ribosome. What is the D arm responsible for? D arm. Binding of tRNA. Very good. What is the which nucleotides are attached to the amino acid binding end? Which nucleotides are responsible? CCA, not ACC. CCA. It's not CAA, ACC, CAC. These are not the one. CCA is the one. Okay. Do not mix it up. Okay, very good. Next one that we want to talk about is the start and stop codons. The mRNA start codons are AUG. We talked about this. AUG is augmentation or inauguration. In eukaryotes, the AUG will code for methionine, which may be removed before the translation is completed. And in prokaryotes, this will code for N-formyl methionine. Basically the same thing, just a different name, N-formyl methionine. And stop codons are UGA, you go away, UAA, you are away, UAG, you are gone. Okay. And N-formyl methionine is responsible for neutrophil chemotaxis. Not very high yield, but you can try to remember this if you want to. Might or might not receive a question, but not high yield at all. The fact that AUG and these are exceptionally high yield, and we have talked about this multiple times. Okay. All right. Are we clear about this? Okay. Okay, let's learn about protein synthesis. Protein synthesis, we have already talked about this in a brief manner over here, right? dRNA, mRNA, tRNA, this and that. Let's talk about this in a little bit more detail. So what happens is in protein synthesis, you have basically, it can be divided into three broad steps. You have initiation, elongation, termination. initiation is basically eukaryotic initiation factors will identify the 5' cap. Okay, what is the 5' cap? Let me see if you guys remember what the 5' cap is. 5' cap. What is the 5' cap? 7-methylguanosine. Very good. Okay. Who has the 5' cap? Who has this 5' cap? 5' cap. Okay, mRNA. Very good. So if the 5' cap was not there, could the initiation factors identify the mRNA and initiate protein synthesis? Yes or no? Okay, so this is a question from your URL. Write it down. 5' cap is, and write down over here, responsible to be identified, responsible for identification by initiation factors. Okay, if you have any pathologies where the 5' capping is hampered, the protein synthesis will not occur, and it will not occur because eukaryotic initiating factors could not identify the protein, could not identify the mRNA. Okay, next one. Next one is the initiation factors will help assemble the 40S ribosomal subunit with the initiated tRNA, and the initiation factor is released when the mRNA and the ribosomal 60S subunit assemble with the complex requires GTP. So what happened over here is that after the initiation factors identify the 5' cap, they will activate the ribosomal subunits in the within the initiated tRNA, meaning that they have identified the microRNA. I mean, they have identified the messenger RNA. Now they have to identify the transfer RNA. So they help assemble the 40S ribosomal subunit within the tRNA, and then the initiation factor is released when the mRNA and the ribosomal 60S assemble with the complex. So they activate the 60S of the mRNA and the 40S of the tRNA. Basically, they make the RNA and the tRNA meet, because only when the mRNA and tRNA they meet can the proper codon of the tRNA form, and that's how the tRNA can go and find the amino acids in the cytoplasm. And the factor that is responsible for doing that is the initiation factor. Okay, and in eukaryotes, the ribosomes are 40S for tRNA and 60S for mRNA. And for prokaryotics, the ribosomes are 30S for tRNA and 50S for RNA. That's that. Just subtract 10 from each of the eukaryotic ones, and you will get your prokaryotic ribosomes. Okay, next one. Next one is elongation. We already talked about elongation. So what happens is, after you have these ones, the tRNA, they go and they bring the amino acids, and the amino acid tRNA binds to the A site. Okay, so this is the A site, P site, and E site. The amino acid tRNA, what they will do is, first of all, they will come and they will bind to the A site. After it binds to the A site, they initiate the methionine, that is the augmenter. The methionine, they work at the P site. Okay, they work at the P site. And what happens is, then you have the elongation. They require an elongation factor and then a GTP. And with the help of the elongation factor and the GTP, they elongate the protein that's being formed over here. Okay, so what happens is, first of all, the tRNA comes and binds to the A site of the messenger RNA, right? And then what happens is the methionine binds to the P site, that is this P site, and then with the help of E site, they keep on moving forward, and then the protein is formed like this. Okay, that's that. The ribosomal RNA catalyzes the peptide bond formation, transfers growing polypeptide to the amino acid in the A site. So, and we talked about mRNA, tRNA. We also talked about rRNA and the ribosomal RNA. I mean, the RNA polymerase, which is responsible for RNA. Ribosomal RNA is RNA polymerase 1, and RNA polymerase 1 is responsible for creating rRNA, and the function of rRNA is catalyzing the polypeptide bond in amino acid in the A side. The ribosomes will advance 3 nucleotides towards 3' end. So they will move at the pace of 3, and they will move from 5' to 3' like this, towards the 3' end, moving peptidyl tRNA to P side. Okay, so this is the process of elongation. Elongation, the only thing that you have to remember over here is that the first binding is to the A, then it's to the P, then it's to the E, and it moves in this direction over here. And the enzymes that are responsible is amino acid tRNA, and rRNA is the function of rRNA is catalyzing the peptide bond. Then they will keep on forming the polypeptide chain until and unless they recognize one of the 3 types of stop codons, that is UAA, UAG, and UGA. When it recognizes the stop codon, they will complete the polypeptide, and they will be released from the hormone. And this process is also an active process, which would require GTP, Okay, so that's that. This is the process of protein synthesis in details. That's that. Okay. We have some questions from the students. First one is from Dr. Sawai. 5-cap is responsible for what? 5-cap is responsible for being identified by the initiation factors. Okay. Next one is, do we have to know the elongation factors in details? No. Just try to remember that the elongation factors, they will identify the mRNA by the 5' cap of the mRNA. Okay. And the elongation occurs by the A side, P side, and E side. And that's that. Any other question? Any other question? Can you guys hear my voice? Is my voice clear? Do you guys have any other questions? Okay. Sir, please repeat elongation factor. Elongation factors are basically factors which will initiate protein synthesis by identifying the 5' cap of the mRNA and the ribosome. And the next one is from Dr. Adano. Translation 5 to 3'. Basically, what would happen is the movement will occur towards the 3' end of the mRNA. The ribosomes will move towards the 3' end. So that's that. Okay. Any other question? Okay. Now, so what would happen? Last thing, are we really tired? How tired are we? How tired are we? Because when it comes to 12:34, 12:30, that is exactly when you guys stop responding. And I keep on asking, are you guys understanding? Is everyone okay? Did you guys hear my voice? And I only get 1 yes and 1 no. That's when I identify that you guys are extremely tired. I know you guys are really tired, but we have some very small portions of the page which is left. Can we finish this portion so that we can start fresh from the cellular part of biochemistry tomorrow? Yes or no? Guys, do we have another 5 minutes left us in order to finish the last two portions? Okay, we're very close. Okay. So what happens? So what happens is after a protein is formed, so we had a what we have is we have a translational modifications. Post-translational modification is that after the protein is translated, there are two modifications which are made to the protein. First of all, trimming. Trimming is basically we remove the N or C terminal propeptides from zymogen to generate mature proteins. Okay, so basically we have a premature protein, which is trypsinogen, and then we activate it to trypsin. And the mechanism by which we do this is removal of N or C terminal propeptides. Now, if you receive a question in your UWorld where they will ask you, how do you activate trypsin from trypsinogen? Your answer should be removal of N or C terminal ends. N or C terminal ends. Next one is covalent alterations. Covalent alterations, okay? Covalent alterations are basically, you can also alter the structure of the protein by adding or removing some substances. Okay. Okay. Wait 1 second. Do we have any question over here? Doctor, I think we missed microRNA. Where did we miss microRNA? Please unmute yourself and ask me the question.
Sorry, did we like cover the microRNA? Because in my book 2019 it's present and we skipped that.
Did we? Wait, let me see. Where was it there? Where was microRNA? Where was microRNA? What was the page number?
Mine it's
not
2019, so it's different.
Page number 19.
No, mine it's 2019 version, so it's different.
I understand. I understand yours is 2019, but which page? At which page did they talk about microRNA in 2019? Because I have 2019 entirely memorized. I don't remember.
In 2019, after intron versus exon. After this page.
Introns and exons. Let me see.
Yeah, it's after intron and exon here.
Oh, it should have been somewhere over here, right?
Yeah.
Right. I think they right. This looked a bit empty to me because I remember this portion. So basically, microRNA, if we have to talk about this, microRNA is nothing that these RNAs, they prevent the production of a particular protein by binding and destroying the messenger RNA that have produced the protein. Okay, so basically what microRNAs, what they do is, they help the cell control the types and amounts of protein that could be made. For example, if an mRNA is responsible for producing a protein that we need, okay, if the mRNA is responsible for producing a protein which we need, then the microRNA can go and not attack the mRNA. Okay, so they will, but if the mRNA is responsible for a type of protein which we do not need, the microRNA will go and they will attack the mRNA. So the protein, so this type of protein is not formed at all. So basically, microRNA is responsible for controlling genetic expression, gene expression. This is the main basic function of microRNA. This is the main basic function of microRNA. If 2020 has skipped out on microRNA, then obviously it's not very high yield. And to be honest, it's not very high yield in the sense that you do not receive a lot of questions from microRNA. But if you have to understand one thing about microRNA, that is microRNAs are responsible for genetic expression. Okay, are we clear about this? Are we clear about this? Okay. Okay. I have been reading First Aid 2020 for so long that I completely forgot about 2019. But while I was reading this, I felt like this part was empty. So that's that. Okay. So if First Aid skipped out on microRNA, then obviously it's not very important. Okay. And you will not receive a lot of questions from microRNA anyways. The only function of microRNA that you have to understand is that it's responsible for protein expression. Gene expression. Okay. Can we go back to post-translational modification? The first one was we had to remove the N terminal and the C terminal end in order to activate a premature protein. Next one is please get hyper me and you. Please get hyper me and you are all the covalent alterations. P for phosphorylation, G for glycosylation, H for hydroxylation, M for methylation, A for acetylation, U for ubiquitinization. Ubiquitinization. So these are all the covalent alterations which you can make to a newly formed protein to give the protein a very unique structure. Okay, so if you want to remember all the alterations, then you can use this mnemonic, because you might receive a question where they will tell you that which of these answer options you will have A, B, C, D, E. They will ask you which of these answer options are not a covalent alteration. Over there, you can have any other method, right? Reduction or what else? Or like any other mechanism, right? Acetylation, methylation, and they can ask you which one of these are covalent alterations, and it should be one of this 6. So it's better that you memorize this. Please get hyper, me and you, because these are all the covalent alterations: phosphorylation, glycosylation, hydroxylation, methylation, acetylation, ubiquitinization. Are we clear about this? Okay. Next one, the last one that we're going to talk about is chaperone. Chaperone protein is not very high yield, but the fact that chaperone protein, wait, let me go back. Okay, where were we? Chaperone protein. Chaperone protein, they're basically intracellular protein which are involved in facilitating and maintaining protein folding. So proteins are supposed to be folded into a complex condensed structure. If you have intracellular protein which can maintain this structure, these proteins are known as chaperone protein. In yeast, heat shock protein HSP60 are expressed at high temperatures to prevent protein misfolding. Over here, if you want to maintain the proper structure of the protein, you want to make sure that nothing happens to the chaperones. So what happens is that in yeast, you have a type of protein which are expressed at high temperature, because yeasts, they have to survive at high temperature. So in high temperature, we know that proteins, they can get misfolded and denatured. So we want to make sure that the proteins in high temperature, they are not misfolded. So as a result, this is just an example that in yeast, you have a type of protein called heat shock protein 60, which prevents the defolding and misfolding of the proteins. And that's how the structure of the yeast is maintained at high temperature. That's that. How important is this? Not very important, except the function. The function is basically they prevent protein misfolding. That's that. Okay, finally we're done. Okay, good. Congratulations, we're done with a major portion of the genetics. Now we will move on to cellular. Okay, are we ready to begin the cellular portion of biochemistry? Why do chaperones not work in fever? In fever, you have high temperature, which can denature the protein. As a result, the chaperones are damaged. So that's why the proteins are misfolded. That's that. Okay. Can we begin biochemistry, the cellular portion of biochemistry, please? Yeast have a very unique protein, HSP60. All other chaperone proteins, all other proteins are not as unique as the yeast chaperone protein. That's why. Can you guys give me 1 second, please? Okay. So okay. What was the okay? Fever. Could you repeat? Chaperone not expressed. Okay. What happens in fever? In yeast, heat shock proteins are expressed at high temperature to prevent protein denaturing, misfolding. What is the question? I mean, what do you want to? Could you please rephrase your question? What happens when fever? What is the question? Are you asking what happens to the chaperone protein when you have high fever? Oh, you already got your answer?
I wanted to know why why we have a denaturation of proteins during fever, but not in yeast.
Because the protein structure in yeast is unique. It's a unique protein structure, because in yeast, what happens is that the heat shock proteins, do we always have a fever?
No.
Okay, but yeasts are always exposed to high temperature. So they have certain protein properties which prevents their proteins to get misfolded. That's why the chaperone proteins in yeast, they can withstand high temperature. So that's that. Okay.
Ah, perfect. So in general, it's a general adaptation.
Yeah.
Perfect. Thank you.
You are welcome. Any other question? If you guys say no, then I will stop. If you guys say yes, then I will explain your questions. Do you have any question, or can we move forward to UWorld notes and emboss notes? Okay. Have we understood the lecture from today? Have we understood the lecture from today? Yes. Okay. Okay. So how about we do a couple of questions from genetics? Let's see if we can associate and apply the knowledge. Okay, let's solve some genetic questions. That would be exciting. Okay, in order for us to do that, can you give me around 5 minutes? Okay, just give me 5 minutes, take a small break, get refreshed. Okay, we only have around 15 to 20 minutes left of the lectures. After this, we will be done and you guys are free to leave. Okay, but since we are solving the genetic portions of the UWorld, if possible, please try to stay behind and see how you do. Okay. Okay, so take 5 minutes off. We will begin at 12:53. Okay. Do we have class on Christmas? Yes. We have classes on Christmas. Okay. Okay. All right. Let's come back after 5 minutes. If a student misses out on a class on Christmas, that's no problem. We will try to provide the lectures for them. But I cannot skip classes on a Christmas, and especially when you guys have already paid for the classes, right? You guys pay by the days and by the hours, and I cannot just go like, we're not going to have classes on Christmas and take money from you. Okay. So we can't do that. And that's that. Okay. So we will be having classes on Christmas. If you guys want, we can cut the classes short, but unfortunately the classes will still be there. Okay. If there's anyone with a problem, and since it's a holiday, I'm sure most of you guys will not be able to be there. We will try to keep the lecture short and give you guys the video so that you guys can watch it. Okay. Does that help? Does that answer your question? Any more questions? Can we do Saturday? Unfortunately, no, we cannot do Saturday. My apologies. Okay. Okay, let's come back after 5 minutes. Okay, is everyone back from their break? Is everyone back from their break? Okay, are we ready to begin the UWorld offline questions from genetics? Okay. Most of the questions which we are going to do right now are associated with the pathology, the pathological portions of genetics. And most of the things, either we have discussed it before or we have discussed it today, but it should be extremely familiar. And I'm expecting out of the 5 questions which we do today, we should be able to get 5 out of 5. And the way we will do this is instead of unmuting ourselves, because what that does is creates a lot of confusion and noise. So what we will do is we will share the screen, and we will have the chat box over here. Okay, for example. Okay. We will have the chat box over here like this. Okay. And if you guys are ready, please provide the answers after you are ready. After I ask you to provide the answer so that it's fair for everyone. Okay, so with that being said, can we jump on to the first question? Can we jump on to the first question? Okay, this will give everyone a chance to participate, and please use the chat box to provide your answers. Refrain from muting yourselves. If you have any questions, stick around and I will explain it for you later. Let's focus on this question over here. Now, number 1, which of the following findings are most likely to be present on further evaluation? We have 90 seconds for this question. These are the answer options. A 15-year-old patient is referred to the physician by a teacher who is concerned about the patient's learning abilities and behavior. The patient's reading and writing skills are significantly impaired compared to other classmates, and the patient often misbehaves in class despite receiving numerous detention. Neuropsychological assessment shows mild intellectual disability. So you underline mild disability and 47 chromosomes. Okay, so we have the student. Is everyone ready? Provide the answer. Okay, what is the answer? Okay, everyone. E. Okay, what is E? What is the diagnosis of this patient? Klinefelter syndrome. Very good. Okay, so 1 out of 5. Very good. Okay, no problem. Next one that we want to go to is this one over here. Okay. Okay. 1 second. Okay. The question is, which of the following is most likely the karyotypic abnormality in this infant? Infant born prematurely to a 42-year-old woman is small for gestational age. Microcephaly, low ears, prominent occiput, and small mandible. And fists are clenched and the fingers overlap, and there's bilateral foot deformity. Clenched fists. Okay, clenched fists, low set ears, microcephaly. Is anyone ready? Okay, what is the answer? B. Okay, what is the diagnosis? Edwards syndrome, trisomy 18. Very good. Trisomy 18. This is Edwards syndrome. Clenched fist is for Edwards syndrome. In Down syndrome, we will have a flat face, abnormal ear, right? Palpebral fissures. Okay. In trisomy 13, okay, trisomy 13, what we will have is What will we have in trisomy 13? Does anyone know? Trisomy 13 is called Patau syndrome. They will have a cleft lip, cleft palate, omphalocele, right? Those things. Okay. Okay. Next one. Next one is this one over here. These findings are most consistent with a Caucasian newborn with facial dysmorphia and cleft palate is found to have a detection or a deletion involving the long arm of chromosome number 22. Okay, so are we all ready to provide the answer? Okay. Okay, so what is your answer, please? Okay, the answer is clearly DiGeorge syndrome or velocardiofacial defect. DiGeorge syndrome. It's not Down syndrome or Turner syndrome because it says long arm of chromosome number 22, right? It says because DiGeorge syndrome is 22q11 disease, right? Are we all clear about this? Down syndrome is chromosome number 21. Syndrome is 45XO. So that's that. Okay. Very easy. This was very easy. So good. So I would assume it's 3 out of 5. Yes. What is your question? Is microdeletion meaning small arm 22q? What is q known for? Small arm or the long arm? Q 22q. We have P and we have Q. Is Q known for the small arm or the short arm? Everyone? Can you guys hear my voice? Okay. So basically P is for the short arm and Q is for the long arm. So if you have a deletion at 22q, it means chromosome number 22 at the location of the long arm. Okay. That is at location number 11. Q is long. Okay. Can we move on to the next question? Okay. Okay. This patient is most likely suffering from which of the following? A 34-year-old man comes to the office due to oral and perioral nodules, okay? Or as we can say, okay, I'm not going to say these, I'm not going to say the term behind this. Let's see who understands. The nodules appeared several months ago and the patient reported that they are similar to the other lesions that were removed 10 years earlier. He has a history of total thyroidectomy for palpable mass. The patient is tall and slender with disproportionately long arms and legs. Very, very easy, very easy. What is the diagnosis? Who is ready? Who is ready to provide the answer? Only one person who's ready, okay. All the other students. So what is the correct answer? What is the correct answer for this one? Does everyone agree with D? Dr. Hussam has mentioned D 6 times. Okay, it's not B. B is Marfan syndrome. Marfan syndrome is not B. It's meant to be. Meant to be. What are the sign symptoms of meant to be? What are the sign symptoms of meant to be? Mucosal acoustic neuromas and marphonoid habitus, medullary thyroid carcinoma, right? That's that. So this patient had a medullary thyroid carcinoma, marphonoid habitus, and acoustic neuromas. Okay, that's that. Last one. Last question. So far, very good. 4 out of 5. Okay. Okay. Last question. Let's see if we can get 5 out of 5 today. The most likely inheritance pattern of this condition is dash, dash, dash. Okay, this is a hard question, so please think twice before providing your answer if you want to get 5 out of 5 right answers. A 32-year-old woman comes to the physician because she's worried that she will go bald. Both her father and paternal grandmother suffered from early onset baldness, but no one on the maternal side of the family is bald. And physical examination reveals a normal appearing hairline without evidence of hair thinning. After reassuring the woman, you explain the genetics underlying the most common form of hair loss in both men and women. Okay. Okay. Is everyone ready? Since this is the last question, let's take some time. Is everyone ready? Okay. Well, what is the correct answer? Let's get the answer right. Okay. C, polygenic. Does everyone agree with C? I'm sure everyone does. Okay, so C is the right answer. So congratulations, you guys successfully answered 5 out of 5 genetic question. Polygenic. Polygenic means there's multiple reasons. The genes are not the only thing. There are multiple other reasons why there could be early onset baldness. That's what it means, polygenic. Okay, polygenic. Are we clear about this? Acoustic nodules. Did we discuss this? No, we did not discuss this yet. Why is it not recessive? It is not recessive because autosomal recessive will only develop in 25% of the offspring. Okay, it is not an autosomal recessive condition because this woman has come to the physician that her father and paternal grandmother suffered from early onset baldness, but no one on the maternal side of her family is bald. Physical examination reveals a hairline. So basically it's not autosomal recessive, because if it was an autosomal recessive condition, it will show that any one of the generation could have been affected, right? So no one in her, no one in her generation has been affected by this. So since no one is affected over here, so autosomal recessive is not the likely answer. The likely answer is polygenic, because in autosomal recessive condition, you have only 25% of the population which gets affected. So that's that. Is it clear? Does anyone have any other question? Okay. Okay. So, okay, congratulations. Thank you so much for answering these questions, right? I am so proud of you guys. Okay, I believe each and every one of you will do really, really well in your USMLE exam, just for the fact that you guys are doing excellent in terms of keeping your concentrations during the lectures. And when it comes to applying your knowledge, you guys are doing really, really well. So thank you so much. And please continue studying like this. And in no time, you will be done with your step 1 with exceptional score. Does anyone have a question? Because if not, then I would like to end the lecture here today, because you will note are almost close to the end. And I'm pretty sure you guys are very tired. I'm barely getting anything out of you guys. Okay. Okay. Does anyone have any other questions? Do we have any students over here who has their exam in 2 months? We received an email from one of our team members, and they let us know that a student has their exam in 2 months, And unfortunately, we could not reply to that email because we were a little bit busy. Okay, does anyone have any exams, USMLE exam in 2 months or 2.5 months? Okay, so if you guys want me to help you out with your lesson schedule, how about we have a small meeting, or we talk about the lesson schedule next week, Monday, after we're done with the class. Okay, so we can come up with a plan, and then we can tell you exactly what you have to study. Okay. All right. So with that being said, hope you guys have a great day today. If that's all, then I'd like to take my leave, and I'll see you guys tomorrow at 9 a.m. Okay. Okay. Have a great day, guys. I'll see you guys tomorrow. Bye-bye.